Cryo-EM conference, and it's my great pleasure to introduce you the first speaker. For today, it's Iskander Husainov from Max Planck Institute of Biophysics, who's going to tell us about the impact of commonly used antibiotics on human gut microbiota. Iskander, please, you have 30 minutes.
Thank you, Yuri, for a nice introduction. And first of all, I'd like to thank to speak along with such great lineup of speakers. And the story I will tell today is very much related to what we were speaking about last couple of days. This is antibiotic resistance. So we all know that this is a huge problem of modern medicine, and the pathogens takes life of millions of people worldwide. And what we need to do, once the resistance is acquired, we need to find new antibiotic. So this is usually done on using the laboratory strains which we can grow, which we can cultivate. And the main criterion, as Yuri mentioned previously, we need to kill the pathogen, but not kill the human cell. However, human cells contain billions of microorganisms which play pivotal role in our health. So what's happening when we take antibiotic? Do we somehow cause a damage of our bacteria? It was largely unknown because we have lack of isolates of human gut bacteria because they are difficult to grow. They sometimes grow in communities, and a recent study from NASA Stipa's group at MBL Heidelberg showed that many gut bacteria are very sensitive to the antibiotics which we use every day. So here is an example of transcription inhibitors, and as you see, all the black squares indicate that this strain is sensitive. So most of the strains they tested were sensitive to the drugs which we use every day almost. And especially, here's the example of tetracycline. So what do we know about tetracycline? Tetracycline is a ribosome inhibitor, and tetracycline has the bacteriostatic effect. So it means that once we put tetracycline together with the bacteria, it stops growing, it doesn't die, and it will restart growing once we remove antibiotic. However, we didn't know how it affects the gut isolates. So we wanted to see why our gut microbes would die from this antibiotic instead of just slowing down their growth. So to answer this question, methodologically, we took these two strains of E. coli. One is laboratory strain, which is well known and characterized. This is E. coli K12. Another one is gut isolate E. coli ED1A. So it was shown that it has bactericidal sensitivity to tetracycline. So we treated this bacteria at two different time points with short treatment 30 minutes and longer treatment of 3 hours. Then we checked how they survive on using microbiological assays. Then we performed the quantitative mass spectrometry to see how the globally proteome reacts to the addition of antibiotic. We also did perform cryo-electron microscopy studies to analyze the ultrastructure of the cells and their subcellular complexes. And we also did the single particle cryo-EM on the main target of tetracycline, the ribosome. So we start with viability assay. The very first thing we did, we checked how do they grow in LB in laboratory conditions. So the growth was pretty much similar. So we used the actively growing cells. We treated cells at OD03, so when all the ribosomes and all the metabolism is the most active. And we took 3 time points, as I mentioned before. So first of all, we checked what's happening if we add antibiotic to the culture. So as expected, we see that without antibiotic, they continue to grow in LB, and the addition of antibiotic stops the growth. Stops the growth, but we don't know whether the cells are dead there or the cells are viable. So then we did the viability experiment. So basically at each time point we washed out the antibiotic and plated them on fresh LB media, the plates. And with this, we could see that the viable cells of lab strain remained the same, while the strain of the gut isolate slowly keeps dying. So like this we confirmed the bactericidal effect of this antibiotic. So then we wanted to see which kind of rearrangements globally on the proteome level happening when you add antibiotic. So for this we used so-called thermal protein profiling experiment. So the idea is that you grow the cells and at particular conditions, then you make 10 aliquots, you divide the cells into 10 exact aliquots, then you heat them with different temperature, and the more you heat, the more proteins will start to aggregate. So then we take the soluble part, label them with TMT, and get the spectra at mass spectrometry. As a readout, we get the so-called thermal stability curve. So this is it's based on the protein stability of the protein stability at different temperature. So if we can read these plots, once the protein is not interacting with anything, is not stabilized, it will be less thermostable, then it will aggregate at lower temperatures. And once the protein is interacting with something, it's harder to dissociate, to degrade this protein with the temperature. So the melting point will increase. So, and this is the difference which we compare and which we measure, and this is our main signal at the TPP readout. So what we saw, so here you can see two strains. In cyan is the lab strain, and in red is the gut strain. In average, we could recover signal from about 1,200 proteins. And what can we read from these curves? So first of all, we see that at the initial steps of the antibiotic treatment, the stability of protein, thermal stability of proteins is changing more than the abundance. So it means that the cell doesn't need to synthesize new proteins or change the protein turnover, but rather it will activate some other pathway with available proteins. But later, at the longer exposure, the cells start to change the abundance of proteins. Sorry. Meaning that maybe new pool of proteins will be synthesized. And the third lesson we learned, and we see it clearly, that once we keep cells in LB for 3 hours, the metabolism is drastically changed. So we see that the abundance of proteins change a lot in both strains. This is reasonable because they go to the stationary phase, and this is completely different state of bacterial life. So taking this in account, we compared the initial response of the cells to the antibiotic. And so out of all the hits which we got, so it was as I said, roughly 1,200 proteins. So we picked those which had big difference in thermal stability. And we noticed that there are many of membrane proteins, and especially efflux pumps in both strains. So if we compare the two strains, how both strains react to antibiotics. So here at the bottom right, you see that the proteins which are more stabilized in the gut strain, and top left would be the proteins which are stabilized in the lab strain K12. So you see that the pool of the proteins is a bit different. And what do we know about these proteins which are getting stabilized or destabilized? So these, they are, some of them are influx pumps and some of them are efflux pumps. For example, protein OMPF is responsible for bringing the nutrients inside the cell. So it's an influx pump. And it functions at the normal conditions. It brings the good nutrients into the cell, and also OMPA is the protein which helps to keep the structural integrity, the double membrane layer of the cells. And once antibiotic is there, the influx pump either deactivate or they leave the membrane, and the efflux pump starts to activate. So if there was some antibiotic which is entering the cell, then the cell trying in response to remove it. And what we learned from our TPP experiments is that in the laboratory strain K12, upon addition of antibiotic, we have low influx and high efflux. So meaning that the cell stops bringing inside but tries to pump out. And the opposite story happening in the gut strain. This is very surprising that the cell cannot regulate this efflux-influx activity, and then it keeps probably influxing the antibiotic inside the cell and cannot get rid of it in time. So then we analyzed the cells using a transmission electron microscope, the strain of the gut strain without additional antibiotic or with addition, and we see that the membranes are largely distorted. And this correlates very well with our hits at the mass spectrometry at TPP, and we could assume that the cell is just not able to pump the antibiotic out. So in this scenario, we would have the following situation, the following. So that in the strain which is bacteriostatic, which doesn't die, just maybe has bit less concentration of antibiotic inside the cell, in contrast to the bacteriostatic one. So this, here I show the preliminary experiment of the master thesis at NASA Stipuslab at EMBL, who measured the intracellular concentration of these two strains of intracellular concentration of tetracycline, and showed that there is about 2 times abundance of the antibiotic inside the gut strain. So then what's going to happen? We assume that it would interact with the ribosome, with its target, and would interact more. And we know that tetracycline is sitting in the A site of the ribosome, so it blocks the ability of A site tRNA to bind, and most probably blocks the ability to rotate, because ribosome doesn't need to rotate in this case. And what we try now to identify is we try to quantify the ribosome states as a readout of the effect of antibiotics. So we want to see how the ratio between translating polysomes, monosomes, and non-translating polysomes is shifted between treated and untreated cells. So what we did, we then went to focused ion beam milling and cryo-electron tomography. So we put the cells on the grid. We vitrify them and as you can see here, these are experimental cells vitrified on the grid. And this, then we need to thin them, because cells, as they are, they will be too thick to pass through the beam. They will be too thick to allow the electron beam to pass through. So we need to remove extra material. So for this, we use focused ion beam milling. So we remove it from the top and from the bottom. So then we end up with a thin layer of cells, and we end up with kind of electron, not transparent, but the thin layer, which we call lamella. So from this lamella, roughly we can get about 7-10 points to collect the cryo-electron tomography data. And when we do, we collect the data using dosimetric scheme, and then we process them using this pipeline. And as mentioned by Misha at the second day, everyone has to find their own way to do subtomogram averaging, because it will not work for every sample identically. So this is our way to do it. So we we align the tomograms in I mode, then do template matching in Dynamo, and then do CTF correction in Nova CTF. And we use in-house Nova STA package for reconstruction and subtomogram averaging. And later, so the step of classification is not yet highlighted because it's the work in progress, and this is not really finished yet. As you can see, there is a tomogram, and you can see inside the cell there are all these small black dots. These are ribosomes, nicely distributed inside the cells, inside the cell. And once we did template matching in Dynamo, we picked these ribosomes mostly in the center, and it mostly picked properly the particles, but didn't find correctly the angles. So we used subtomogram alignment with Nova STA to align the particles. And so this is the first reconstruction. So we got the ribosome reconstructed, and if we back project it to the tomogram, we see like the distribution of the particles inside the cell. And here we can see the 3 tRNA bound. So this is an average of all the ribosomes present in the cell in particular moment. So this will it will contain like all tRNAs and some factors. So what do we learn and what do we see from this from these structures? So first of all, we can restore, or we can resolve some flexible regions which are involved in translation in situ. So the cell is performing translation. So we can see, because they are active. And like this, we see the L stalk, the large subunit. We see the part of S1 protein. We see some particles showing the expressome involvement with RNA polymerase. And also we saw really strong density at the entrance of the A site near the shoulder. So this most probably corresponds to the density of tRNA, which is being delivered to the ribosome. So we would assume that this is the time limiting point when the ribosome has to discriminate between different tRNAs. So it will be slow inside the cell, and we would see the big average of this. Also, we could attribute this to EFG density because they have similar binding sites and kind of similar shape. And if we assume that this is an average, so we will need to classify it further to understand what it is exactly. And lastly, we performed single particle cryo-EM. So why do we do this? So we know that antibiotic binding the ribosome. So maybe this is the ribosome property, that the ribosome would be look different, or it would be somehow more susceptible to the binding of antibiotic. And so we purified the crude lysate. So the idea was also to see how much we can sort out, how many states of the ribosome we can sort out by purifying them in vitro. So we took the whole ribosome pool from the cell, put them on the grid, and so this is average structure of this single particle analysis. So it went pretty high in resolution, and we see that there are, like in the average, we see 3 tRNAs bound there, and the quality allowed us to see the ion metals quite well in the density. And then we did some 3D classification, multiple rounds of 3D classification, and the idea was to see how many states we can recover. So these are the the most pronounced states, the difference between most pronounced states. So this is a rotation of the 30S, and the presence of and absence of tRNA was also detected, and the movement of helix 69. We didn't see many peripheral regions would do defect and we didn't see the factors bound because they most probably dissociate during purification. And then we looked at the antibiotic binding site. So the antibiotic binding site was absolutely identical. So this is not the property of the ribosome that it binds somehow the antibiotic strongly or not. And so the binding pocket is exactly the same and the sequence is exactly the same there. So we would can conclude that this is mostly the property of inability of cell to pump the antibiotic out. With this, I would go to conclusions. So we see that the effect of antibiotics is different than that we know from laboratory strains. So our gut microbes may react significantly different. And using Cryo-ET and proteomics, we combine these two methods to see the global response of the proteins, and we characterize the ultrastructure at nanometer and sub-nanometer level. So for future, I believe that combining and joining the forces of multidisciplinary techniques, we will be able to understand a bit more how the antibiotic how we can efficiently modify the antibiotics and how we can use them to kill indeed the bug, but not disturbing our own gut microbiota. So with this, I would like to proceed to acknowledgements. I'm very thankful to the group and to Martin Beck, who allowed me to do this study in his lab, and great thanks to Natalie and Beata, who helped in proteomics data analysis and Cryo-ET data analysis, respectively. And Matteo, Patrick, Shamal, Christian, Andre, and Max for continuous discussions on cryo-electron tomography, on ribosome. Of course, many thanks to the EM facility of Max Planck and of EMBL, where I started this project, and to proteomics group, to Julian especially, and special thanks to Misha and Nikita, who helped with template matching and with dealing with Dynamo, and our collaborators from NASA Stipes group, who basically found this pronounced effect of antibiotics and let us discover it more. And many, many thanks to organizers who managed to deal with this combined hybrid version of conference. This is very first time for me, and I'm very happy to present personally, but if I would be online, I would not feel much difference because you feel you are around here. So thank you very much for your attention.
Okay, do we have any questions? Misha.
Hi, Iskander. Beautiful work. I just want to ask you if you already can say something about how much the structure and the distribution of ribosomes into classes is different inside your cells treated with an antibiotic compared to the purified ribosomes.
Yes. Unfortunately, I cannot say it right now because I didn't do classification yet. So this is the work very much in progress. So we are now exactly at the step when we can classify, and when the most interesting part will come on the stage and the most exciting time. So hopefully in a year I will be able to answer you.
More questions? No more questions. I have a question. So as far as I understand, so you have two strains of E. coli, right? So in one strain, the same antibiotic, tetracycline, is bactericidal, in the other, it's bacteriostatic. So, and you did this thermostability test for the proteins. I mean, so I cannot say that I quite understood. I mean, what exactly you were trying to look at? I mean, so by doing that experiment.
So first of all. No, not first of all, the main, that since the antibiotic has different effects, so it means that the cell will react differently, and probably if the cell starts to die, it will start to try to induce other proteins and to make other proteins busy, so active. And by this experiment, we can see which proteins are more activated in one strain in comparison to the other strain, and we can see inside the same strain before adding antibiotic and after, which kind of, so proteins get stabilized, means that which kind of pathway will be induced and so on. So, and in mass spectrometry, yes, we have many hits, and we need to carefully analyze the data because you need to find the correlation between them, of course.
So it's too soon to say basically what is actually different between the two strains, so that makes one bacteriostatic, and the effect of the antibiotic bacteriostatic in one and bactericidal in the other.
So for us, for the moment, we see the best hits in the inability of the bacteria to remove antibiotic from the cell. So the efflux.
So it's not the on-target effects. It's as much as the ability to pump it out. I mean, I'm not sure what.
Most probably, yes.
I see.
Yes.
Makes sense. Let me check the chat. So it looks like we don't have any. It looks like we don't have any questions in the chat. So let's thank Iskander, and we'll have 5 more minutes before the next speaker. Okay, so let's move on. So it's my pleasure to introduce, can you hear me?
I can hear you, yes.
Yes, so it's my pleasure to introduce our next speaker, Werner Kuchelbrand, who's going to tell us about the high-resolution cryo-electron microscopy of membrane protein complexes. Werner, please.
Thank you very much, Yuri. Thank you for the introduction, and thank you, Olga, for asking me to speak at this really excellent meeting. It has been great so far. Enjoyed it tremendously. And what I will focus on today is our work on membrane protein complexes, and these have been in the center of my interest really for the last 40 years, ever since I started my postdoc in 1981. And so we've seen great progress, especially in the last 10 years, as you all know. Initially, we needed to, crystallize things, look at 2D crystals to get to high resolution. But that is no longer necessary. And now we can get to as high and even higher resolution without any crystals at all. And this is remarkable. It is really due to a number of things, mainly these new detectors, which you heard about from Richard Henderson on day one, but also software, very important. And so we now can, without crystals, get to high resolution by Cryo-EM. So here's a quick outline of my talk. I will talk about 3 complexes mainly. First, the chloroplast ATP synthase, then mitochondrial complex 1, and then mitochondrial ATP synthase at the end. So this is a slide that's now already 7 or 8 years old, and it was a commentary that science asked me to write when the first of these structures appeared, that were at the time 3.2 angstroms from single particles without crystals, and that was unprecedented. And it marked really a revolution in the field in structural biology, indeed in the whole of molecular cell biology. And this is still going on. As you know, these are 3 of the first structures, the large ribosomal subunit of mitochondrial ribosomes, TRPV1 channel, and this is our own FRH complex, is our favorite standard test specimen. It's we use it, have used it in preference to apoferritin because it actually does interesting chemistry and just finds some interesting cofactors. So this work was really the work of Derek Mills and Janet Fonk, and Derek unfortunately passed away almost a year ago, last July. Many of you would have known him. He was really the heart of the department, and he took immense pride in keeping the microscopes in top shape and getting the best results out of them, and including these high resolution structures that I will show you. And but literally until a few weeks or even days before he died, he was writing this review, which is now available online, and Quarterly Reviews of Biophysics, and it's his legacy, if you will. And anyone who's trying to set up an EM laboratory, and even old hands, can learn enormous amount from his review, where he puts, again, 40 years of experience into this one review. So it's highly recommended. So, as I said, Derek wouldn't rest until he got the best out of his machines, and with our FRH complex, he got a map which I would normally call 2 angstrom map, but Derek minded passionately to go below 2 angstrom, so it's 1.97. And I know if you had lived to see the day, he would not rest before he gets to 1.2 angstroms with this complex. That is probably not going to happen. But what you see is here the 12. This is a complex with fairly high symmetry, 12-fold symmetry. So it makes it easier. What you see is these red beads, and the red beads are water molecules on the outside of the complex, and each of them is even fitted with a water molecule. And this is really one of the hallmarks of a 2 angstrom structure. So I told you the interesting cofactors and chemistry. So here are some of them, and there's in the lower right, there's a nickel ion center, and I have a little movie of that that shows you the exceptional quality of this map. And there's the iron, there's the nickel, there's numerous water molecules. And so this is really exemplary of a 2 angstrom map. Right. Now I'm going to go to energy converting membrane protein complexes. You all know this is a textbook image from Bruce Albert's great book. And it shows in a nutshell how this works. So you have 2 membrane protein complexes, one that works as a proton pump and another that converts the proton gradient and uses it to make ATP. And this is really in a nutshell how biological energy conversion works in chloroplasts or in mitochondria. In chloroplasts, you have membranes, 2 membrane systems, actually 3. It's the internal thyroid membrane. You have an enclosed compartment here, which protects the proton gradient set up by these reaction centers. And then a central part of it is the ATP synthase here, which converts the proton gradient, uses it for making ATP, as I've said. So these electron transfer complexes, we knew quite a lot about from crystallography because they're solid state devices and they're fairly rugged, and not all of them, but some of them at least. But we did not have a structure of an entire ATP synthase. So when these direct detectors came along, this is one of the things I was keenest on, to use these detectors to get structures of intact functional ATP synthase, which have not been available until that point, because simply they don't crystallize without falling apart, or even if they do, the numerous very similar states, they would all be in the crystal lattice, and then you would get an average structure. And this is, of course, not a problem you have in Cryo-EM. So Alex Hahn, when he was a postdoc, he got, so the world is full of chloroplasts, you can easily make large amounts of this ATP synthase. So Alex got himself a couple of kilos of spinach and proceeded to isolate the ATP synthase, which he did, and so showed it is pure, and it's complete, and it's active. I won't go into that, this is all, you can read up about it if you're interested. And he did Cryo-EM with, here is an image. The image you can't recognize very much, but if you look at the class average, you can actually see there's these long helices. This is the peripheral stalk, and in the rotor here, rotor assembly in the membrane, this would be the membrane part. You see these thin white lines. These are also alpha helices of the rotor assembly. So this is the three different states. One of them is by far more popular than the other two. And this is therefore also the highest resolution. You can tease this apart further by mask refinement, and the F1 catalytic part, we got to about 2.9 angstroms, and the rest also quite good, 3.4, and overall makes 3.1 angstroms. So this is now a good 3 angstrom map. This is what it looks like, colored by subunit. You can see all the side chains very well. I won't go into it in great detail to give you more time for the other things that have not been published, or at least not been published for so long. Right, so the other great thing about electron microscopy, as you've just heard from Iskander, is you do tomography and you can find out where these complexes are in the membrane. So this is here now artificially colored, of course. Light green is the stromal membranes, dark green is the granular stacks, and the ATP synthase F1 heads are these little yellow spheres. And as you can see, they're fairly evenly distributed in the stromal membranes and also on the granular end membranes, but they're not in between the granular stacks because simply they don't fit in, they would be squeezed out. So this is a nice result from Bertram Daum when he was a student and then a postdoc in the lab. Now let's look at mitochondria. Same principle. You have proton pumps that pump protons across a membrane, and then you have the ATP synthase that converts the mitochondria synthase, that converts the proton gradient and uses it to make ATP. Same story. We have electron transport proteins, the respiratory chain, where we had X-ray structures. We now have a much better EM structure, as you will see, also from Leo Sazanov's talk later on today. But we did not have a good idea of the synthesis. We knew how the catalytic part worked, but we didn't really know how the membrane part worked because there were no X-ray structures. So this is then the respiratory chain. It's in the crystal and a series of 3 proton pumping complexes. Let's look at complex 1. And this is a collaboration with Volker Zickermann and Vivek Sharma and others in Helsinki, and Christian Paré, the person who did all the work. So he got a 3.2 angstrom map, which we were very pleased about, published a couple of years ago now. And this, again, is colored by subunit. There's 43 subunits. It's quite a complicated assembly. And it has this large peripheral arm which protrudes into the matrix. This is water accessible, and the membrane part, which is not water accessible, and sort of sits in the lipid bilayer of the inner membrane. So again, with now best microscopes and best cameras, which and using a much smaller pixel size, Christian got a 2.1 angstrom map. And again, you see in the water accessible part, you see these red beads. These are water molecules. In the membrane parts, you don't see the red beads on the surface, but there are lots of them on the inside. And if you look inside, this is a section of the map on the inside of the complex, and you can fit this with a polypeptide and water molecules. So the inside of this thing here is really full of altogether about 100 water molecules, and you need this to transport protons from this part to where they leave at the far end, as you will see. So here's again some details of the map. This is a very good 2.1 angstrom map, and you can see the water molecules. You have pointed out with the red arrows. Here's some more and more. So altogether about 100 on the inside, and so how does it work? What does this tell us about how it works? So the truth is, we still don't know. Maybe Leo knows, and maybe he'll tell us about it in his talk. But for us, although we're getting closer, it's still not clear how the whole assembly works and why you need such an enormous 1 megadalton complex to carry out these reactions. But so there's a channel here, actually 2 channels. One we call the E channel, and it goes down to this, links up to this water-rich, cavity in the inside, and the Q channel through which the quinone enters. The quinone is water lipid soluble, so it lives in the membrane. It binds there, and then it's reduced to electrons that come down this path of 8 iron sulfur clusters, and then 2 protons join, and then you have reduced quinone, and that leaves. So that's one side of the story. That's the quinone chemistry. But then there's a whole other side, and that's the proton pumping. And what we still don't know is how the two are coupled. But what it appears to be is that you have another proton inlet here. This was discovered by theoreticians we were working with, Vivek Sharma in Helsinki. So the proton inlet residues, and they seem to let in a couple of protons here. And then this links up to this long water channel, internal water channel. So they then go to the other end and they pick up another 2 protons from the matrix. And then they leave, the exit is here. There's also this, Leo Sazonov published his first last year in his great science paper on the ovine complex. So there's one let out, there's apparently 4 ways in for protons, but only 1 way out. And then how this is coupled to the quinone chemistry, we still don't know, at least we don't know. But there's some interesting things going on in this region here, which is at the interface between the peripheral arm and the membrane arm. And we have 2 structures, one resting and one active. And if you compare the 2, you see there's loop movements going on. So there's iron salt bridges being broken and forming. And this is probably the crux of the coupling mechanism between the quinone chemistry and the proton pumping. But the details, we don't know yet, but we're getting very close. So in the meantime, we've also done complex 1 from other organisms, in particular plants and algae. Plants rely, of course, on chloroplasts, as you've seen, mostly for their energy, and algae do too. But they also have mitochondria, and the mitochondria are important, and you can isolate complex 1. And we've done this work in collaboration with Hans-Peter Braun in Hannover. He spent years, 10 or more years, isolating complex 1 from plants, and found that they have an interesting extra domain, which is shown in yellow and orange here. That's the carbonic anhydrase domain. And then also we discovered there's a bridge domain that links the peripheral arm to the membrane arm. And here at this bridge domain, there's 3 subunits, and this bright pink subunit is a ferredoxin. It's found out by Niklas Krusch, who did this work by comparison to other ferredoxins. So it's a bit unexpected. And again, this similar story in Polytomella, which is a chlorophyll-less green algae, so it depends on its mitochondria completely. And it also, lives in drying out mud puddles. So its complexes are very, very robust and quite rugged. And this is extra subunits, 51 altogether. And here the ferredoxin is actually embraced by one of these supplementary subunits, so it can't leave. If you look at the yeast complex I've just shown you, these 2 subunits, shown in dark pink and red, they are there, but this one is missing. So we don't know whether it's just dropped off or it's meant to be there, it's come off. It's not known. But the interesting thing about this carbonic anhydrase is, Niklas was able to resolve a zinc ion in Arabidopsis, which means that this assembly is active in Arabidopsis, but the zinc ion is not present. There are actually no ions in the active side. So that means this assembly is inactive. And it doesn't need to be active because Polytomella is, as I showed, it's chlorophyll-less algae, so it doesn't do photosynthesis. Arabidopsis, a plant, of course, does. So that's nicely summarizes the difference between the 2, this ion here. And yeah, there it is. So now, for the last few minutes, let's look at the ATP synthase. So I've shown you the chloroplast synthetase is equally distributed, is monomeric, but in mitochondria that's not the case. The ATP synthetases are dimers, and we know this from, again, from tomography, and here is a render tomography volume done by Bertrand Daum already about 10 years ago, and it shows these rows of dimers along the ridges of the inner membrane crystal. And this is a common feature. It's a hallmark feature of all mitochondria. We've looked about 15 different species, and they all show this in various forms and various extent, but they're always dimers, and they always form these rows. So we got hold of these dimers. This is a fungal mitochondria. We again went to polytomella for the reasons I mentioned. It's quite robust and quite rugged, and this is borne out by the particles here of Nicholas Klusch and also complex 1 work on plants, and Bonnie Murphy. They collected 730,000 particles, and of these almost 390,000 were good, and that's very unusual because mostly in single particle work you get at best 20%, usually around 5%, and the rest is damaged somehow by the airborne interface. It's not the case here. So that means this is a really quite a robust, rugged complex, and it's helped us greatly to collect a great number of particles. And then Bonnie got to work with it, and she was able to take this to 2.7 angstrom resolution. And then, amazingly, she was able to separate by image processing, classified refinement, 13 different rotary states. And if you put these all together, you get, they tell you how this works. So this is now, again, the map colored by subunit. It's quite unusual for an ATP synthase. It has this very rigid and robust looking peripheral stalk, whereas in chloroplast it was, if you remember, it was just two long, thin alpha helices. Here it's really, it's 10 subunits, all designed to keep this very stable. So here are the 13 rotary substates. There's 3 main states, as in chloroplasts, but now Bonnie was able to separate these into 6, 4, and 3 substates. And they all look very similar, but if you look at where for instance, in red here, the ATP in the active site is there in 2 of these catalytic beta spins, but not in the 3rd. If you use that as a marker, you can actually see how this rotates around. And you put these are like stills in a movie. You can put these together, and then you get a movie. And this is the central drive shaft, the gamma subunit, and it rotates. The F1 head in light and dark green rotates with the drive shaft for about 30 degrees, and then recoils and relapses, and then the next power stroke starts. And this is how ATP is made. And on the top is this subunit which connects the peripheral arm here in gray to the top of the F1 head. And that's like a universal joint, and it's flexible, and it has this single polypeptide link between these 2 domains, and that makes it flexible. And it needs to be because you have a mismatch in the symmetry between the 3-fold F1 head, where the catalytic reaction goes on, and the motor, which I'll show you in a minute, in the membrane, which in this case has 10-fold symmetry. So to equal out the mismatch, you need this flexible joint, and this is completely conserved, so we're convinced that the same mechanism holds true for all ATP synthetase, whether in bacteria, chloroplasts, or mitochondria. So this is the whole thing. Inside you see the halting rotation of the central stalk of the F1 head in green that goes with it for 30 degrees and then relapses. And up here, the universal joint that flexes around this single polypeptide chain. Right, so this is in the membrane. Here is the motor. Let's have a look at that. And the motor is, again, it's an amazing assembly. So this is the rotor of 10 C subunits, and almost at right angles to these long alpha helices, which we discovered already in the 6th angstrom of this assembly. And this was quite unexpected again, because until then it was thought that this subunit has helices just as every other membrane protein that spans the membrane just once, and it's about 40 angstroms. So these are 80 angstroms, they're twice as long, and they're roughly at right angle. And this assembly is the core of the motor of any ATP synthase, and it makes a little window here, and through this window the protons go in, and they come out there, and then this drives the rotor and drives the machine and drives the ATP synthesis. And this is pretty much the same in mitochondria, shown here on the left, and chloroplasts. We have the same distance of these two half channels, one from the acidic lumen of the crystal or the thylakoid lumen in chloroplasts, and then a wider funnel of the leading to the matrix, where the pH is around 8. Whereas in the thylakoid lumen, it's around 6. So you have a great pH gradient which drives this rotation. It's shallower in mitochondria, but it's nonetheless there. And the principle of driving this rotor and making ATP is the same in both cases. So this is how the machine works. So the protons come in, protons are now in blue. They come in, they protonate this glutamate. This is drawn by Alex for chloroplasts, so there's 14 of them, 14 subunits in the rotor. They come in through this inlet, they protonate the glutamate. The glutamate partitions into the hydrophobic part, which is light green here. The rotates round. When they encounter the exit channel in light blue, they're connected to the high pH of the matrix or the stroma, and they diffuse off. And this process drives the rotor and drives ATP synthesis, and is really what keeps us all alive, you can say. And you can now draw a little drawing, and like a model of the dimers here along these ridges in the membrane crystal. This is the complex 1 from polytomella. So both are from the same organism. This is polytomella, this is polytomella. And you can now see how the protons are pumped in through complex 1. It's actually 4 protons for each couple of electrons transferred. They diffuse around in this not quite confined volume. It's open on one side, whereas in chloroplasts, of course, it's closed off, which makes life easier. But here, it's a semi-confined volume, and they diffuse probably mostly along the membrane surface to the ATP synthase, where they drive this rotor and make ATP in the F1 part. So that brings me to the end of my talk. I need to thank a large number of people. Derek, of course, who unfortunately is no longer with us. He was behind a lot of the high resolution work. He kept the microscopes in top shape, and really owe him a great thanks. And together with Janet, who did a lot of the image processing and still does a lot of the image processing, including all the processing on complex 1 that I've shown you. Chloroplast ATP synthase is really work by Alex Hahn, again together with Derek Knowles and Janet Fong. And Thomas Meyer, who is now a professor in London, he started the whole thing before he left. Christian Paray has been instrumental in working on Eurovia Complex 1 together with Volker Zickermann and Vivek Sharma. Volker is the biochemist. He provides the preparation. Vivek is the theoretician. He discovered this proton inlet I've shown you. Redoxis Complex 1 is Niklas Klusch, together with Hans-Peter Born and Jennifer Zenkler in Hanover. Polytomera Complex 1 is really all Niklas's initiative. He just went on from the ATP synthase dimer, which he isolated and thought, well, rather than pouring the complex 1 into the drain, he thought he'd look at it in the microscope, and it was a great success. And polytomera ATP synthase, I've shown you, is the work of, again, Niklas, who did the biochemistry, Bonnie, who did all the image processing, and with the help of Özkan, fitted the model, and Julian Langer, who did mass spectrometry to show which subunits were there and what their sequence was. Right, so this is the department, our retreat. Again, it's about a year ago. We were already in the pandemic, so we were all wearing masks like good citizens, but not all the time. So here the masks are off, and you can see some of the people. This is Alexander who did the chloroplast. Here's Niklas, who did the great work on both the ATP synthase and complex 1 from polytomella. Behind him is Bonnie, who did this image processing tour de force. This guy you know, and there's a number of others, more than I care to mention individually, but they're all great and doing wonderful work. So thank you very much. That's me at the end, leaving time for discussion, I hope.
Werner, thank you very much for... Werner, thanks a lot for for sharing such amazing structures. Actually, I need to say, I caught myself while listening to your talk that when I was a student back at Lomonosov Moscow State, and we were studying all the molecular biology, so all the proteins that you showed us, we were drawing with rectangles and ovals.
Right.
And now it's so kind of unbelievable, I mean, to which level you have resolved them. Do we have any questions in the, in the audience? Misha?
Werner, hello, it's me, Misha.
Hey, Misha.
Yeah, long time no see. So I actually never asked you this question. I wonder, so you have, on the complex 1, you have 4 entrances for protons, and on the 1, which is where 4 protons exit. And it's really far away, like on the molecular scale. Why would you think that it has to be so far?
I really have no idea. Perhaps Leo is giving a talk later today. Perhaps he knows the answer. I think it's probably more efficient if you have only 1 exit. That's the only reason I can think of. I mean, once you have protons in this internal shell, which is full of water, it doesn't really matter how far. And they jump from one to the next by the Grotius mechanism. So it doesn't really, they will travel, you know, in bulk water, they go a very long way in a very short time. So I think that's not in itself amazing. It's probably just preserving some sort of energy by having only 1 outlet, but I cannot answer for sure.
More questions? I don't see any raising hands. I have a very naive question from a person who is very far from the membrane biology. So is it difficult, I mean, to solubilize all these proteins for the Cryo-EM studies? Is it any way similar to how you prepare them for crystallography, for example?
Of course, it's difficult. Otherwise, it would be doing it. No, it's in many ways it's very similar. So you use detergents, and you have to be careful which detergents you choose because some take them apart, others don't. Some detergents are very good, others are not very good at all. And also what you can do, which I haven't talked about, you can put them into lipid nanodiscs. So you can run a ring around them, a polypeptide ring, which is then filled with lipids. And it's like a mini membrane around the membrane part. And that is what people do. Sometimes it works well, sometimes it works less well. And what I haven't said, the chloroplast ATP synthase was in exactly such a lipid nanodisc, whereas the other ones weren't. So there's various ways. And of course, none of this is an option you do not have in crystallography. Yeah? So they will not crystallize these nanodisc protein complexes as far as I know. So it's similar, but it's different.
Thank you very much, Werner. Thank you.
Right.
All right. So we're going to have a lunch break right now. And I just wanted to remind everyone so that we have break until 2 p.m., 14:00. Oh, yes. And I was also asked, I mean, to tell everyone that there are some snacks available in the coffee room. Okay. Okay, colleagues, let's resume our session. So, can you hear me? Can you hear me?
Barely.
Okay, so I would like to introduce Abhay Katesha, who is going to tell us about thermoscientific selectors together with Falcon 4, enhancing the power of Cryo-EM. Abhay, you have 30 minutes, please.
Okay, thank you very much. I hope you can hear me. If you don't, just shout. And I hope you all had a nice lunch. Thank you very much for inviting me to this wonderful conference. I have been seeing some fantastic talks since the last 3 days. I'm going to show you some of the work that we have been doing, especially with Selectris imaging filter, Falcon 4, and I'll also show you some of the results that came out from the combination of our CFAC Selectris and Falcon 4 together. So electron cryo microscopy has really started dominating the field of structural biology since last few years. Before 2013, there were like a handful of structures which were going sub 4 angstroms. But nowadays, every week we are seeing structures going to higher and higher resolution. And this is happening because of a continuous development that's happening in the hardware side of the microscope, but also on the software side. Software in terms of data collection, ease of use, and also some fantastic development in data processing side of things. And all this had led to this explosion of structures from Cryo-EM. This is one of the slides that I really love, and I always show this. This is the microscope at Tokyo University, where in 2018, Rado Sindanao showed this wonderful structure of apoferritin. You heard about apoferritin in previous talks as well. Holger Stark gave a wonderful talk showing atomic resolution with apoferritin. But in 2018, for the very first time, Rado showed this 1.6 angstrom structure that you can achieve with Cryo-EM. That was, I think, the first structure of a biological sample going to such high resolution. And on the right here is another structure of apoferritin, which I did on the same microscope when I was in Tokyo, with just 35,000 particles going to 1.7 angstrom with about 8 hours of data. So just fantastic that what you could achieve on a test sample. Now this is like a test sample, rock solid. So yes, we can achieve good resolution. But if we look at many other structures, mostly many membrane protein structures, as well as flexible proteins, most of the time these structures are stuck to 3 to 4 angstrom resolutions. It's still very good. And since about a year and a half, we are seeing this shift where structures are moving towards higher resolution. We are seeing sub 3 angstroms, and in some cases, we are also starting to see sub 2 angstrom structures of such challenging molecules. So what do we need to get high resolution? The very first thing is sample. Of course, we all need good samples. If we throw in the garbage, we will get the garbage out. That really stays here. But we also need good support films. And there has been quite a few development in this area, in the sample prep and the support film area, where we have now this wonderful gold grids from Lori Passmore and Chris Russo. Also this AU hexafoil with the small holes coming from Russo group. There are affinity grids coming from Bob Glazer and colleagues, which are really helping as well, solving crucial problems. We have graphene and graphene oxide grids. So lots of development is happening, which is kind of helping this push. We also need stable microscope. Lots of hardware and software developments are happening here. But we also want high throughput data collection nowadays. Just a couple of years ago, and still today in some cases, people have to collect like data for like 2 days, 3 days, sometimes even 2 weeks, and hundreds of thousands, even millions of particles to get to some decent resolution. So we want to reduce this time. to few hours, if possible, to achieve such resolution. And that also falls into the last point. We need a fast, sensitive detectors with the highest DQE, so that with less amount of particles, you can achieve higher resolution. So what we have been doing in this area is, if we look at the standard way of data collection, we look at this holy carbon foil, we collect some images here, and then we mechanically move our stage from this hole to this hole. And whenever there is a mechanical movement, we have to wait for the drift to settle. And this drift waiting time can be between 10 seconds to 30 seconds, depending on your setup. If we just count 10 seconds wait time, then reaching this 21 holes, we are going to lose 210 seconds just for waiting. So we need to do something over here. And this is what was done with optical effect. Instead of moving your stage, one could just move the beam and use beam image shift to try and reach all your neighboring holes. And when you do this, you can reach all these 21 holes with just 4 stage moves, effectively losing just about 40 seconds. So that gives you a good improvement in throughput. But doing image beam shift, you get beam tilt. It's an aberration. You get a coma, which can restrict your high resolution information. But using this method called as aberration-free image shift, which is already integrated within EPU software, you don't need to worry about it. It corrects your coma, it corrects this beam tilt on the fly, so you still get a high resolution, high quality data. This is how it looks like. This is now a real sample. This green, dark green holes are the way we move by stage shift, and the cluster of colors we move by image beam shift. This is just showing a 6 micron beam shift. You can do like a 12 micron, 14 micron, and your throughput keeps increasing. And in this case, we could go through this entire grid square, 243 holes, just with 18 stage shifts. So tremendous improvement in your throughput. In addition, what people have been doing was they had to have a very large beam in order to avoid these fringes. So this is your hole, your detector or your field of view is really small, but your beam has to be this giant. The reason for this is because of these fringes. These fringes are diffraction coming from the C2 aperture, the edge of your C2 aperture. As a result, you need a giant beam. But by focusing the C2 aperture in your sample plane, what we can do is effectively remove all these fringes. So instead of now 4 images within your hole, you can do 6 images within this hole. This is a 1.2 micron holes. On a 2 micron holes, you can do even like 10 images, 12 images. So you can really efficiently shrink the size of your beam to the size of the field of view that you have at the magnification, essentially giving you much more area to collect your data. And other improvement was this Falcon 4. Now Falcon 3 was very good. It has the highest DQE. However, it was slower. It was taking 60-second exposure time to get 1 image. Falcon 4 runs 10 times faster. So instead of that 60-second, now you can get the same image with the same quality, but just within 6 seconds. And based on your setup, you can change this. You can use up to even 2 seconds. I have even used data collected with 1 second exposure time, and you still get wonderful images. So when we combine all these things, we can start to do some real good science and start to answer some real biological questions. So this was all about promoting these tools. Let's now look at some science. And then all these tools are really wonderful. Like it allows you to do these things. So this is a collaboration that I set up with Radu Arasescu at LMB in Cambridge, where Radu is interested in looking at this GABA receptor. GABA receptors are the neuronal controlling neuronal synapses, neurotransmitters. They are about 200 kilodalton human membrane proteins. There is one crystal structure which came after decades of work in Radu's team, the homomeric form of GABA, which went to 3.2 angstroms. This receptor is very important because any mutation in here can lead to several diseases. All these neuronal diseases are responsible through this receptor. And also it's a major drug target. But one of the biggest problems is heteromeric receptor does not easily crystallize. And even if you get some crystals, it doesn't differ to high resolution. And this is where Cryo-EM comes in. And we use all these techniques that I showed you earlier. And with just 12,000 images, 1,200 images, we can achieve 2.6 or even 2.5 angstrom structures with a throughput running at 200, 300 images per hour, depending on the grid type and the setup that we use. So then in just a few hours, you can have a full data set, which can give you to such high resolution. Now, this was the very first data set that we collected almost 2 years ago. Nowadays, with similar setup, I can even run it like 400, 500 images per hour and still get such a wonderful quality. And you can see this very nice, all these drugs that are bound in this binding pocket. You can also see how they are interacting with water molecules. So that's a one thing that this is helping. But then the next question we ask is, can we push the resolution even further? And also, can we show this on all the different samples, more challenging molecules? Well, here, just to push resolution, just keep adding more and more data, more and more particles, it's not going to work. It's not going to give you high resolution. And I'll show you why in a minute. What we need is, we really need high quality images. We need high signal to noise ratio in our images. And this is really very well explained by Peter Rosenthal and Richard Henderson here. Wonderful paper from 2003. If Richard and Peter are looking at this, apologies, I'm trying to explain this. If I make these mistakes, apologies in advance. But this very nicely elaborates this concept. We want to improve our resolution. And if we want to do this, we need to increase this term down here. To increase this, we can either increase this n is our number of particles. So more and more particles you add in, your noise reduces and your resolution can improve. But this works to some extent because then the structure factor starts to fall off. Other term is B, which is your B factor. It's fall off of your amplitude as you start going at a higher and higher resolution. And Holger did some explanation at the start that lower B factor is better to achieve high resolution with less particles. However, if your B factor is very high, then no matter what you do, you can double the number of particles, triple it, quadruple it, you will not see any difference in your resolution. You just start hitting that wall. Only way to do it is just improve something in your data collection, improve your detection quality. And again, this is the plot from this paper here. If your B factor is about 1000 to get to 5 angstroms, you will need like millions of particles. But if the B factor can be improved to 500, then one can get to 5 angstrom with just about slightly over 100,000 particles. If you can improve it to 250, then you need less than 1,000 particles, and at 125, less than 100 particles to reach 5 angstroms. So it plays very important role here. So how do we improve further the signal in our data? One way to do this is by using energy filter. So this is now EELS spectrum on ice and some of the samples that you will see later. And what you're seeing is that this is your signal here, and this is a plasmon. This is the electrons which have scattered inelastically, which do not contribute to your signal, but forms a part of noise. So we want to remove this as much as possible. One can remove this by filtering it using a very narrow slit. Here I'm showing the 10 at 10 eV slit width, so 5 on each side. You can essentially remove all this noise and your signal can improve. One of the problem is the filters, and many of you who are using microscope, you know that filters are very unstable and it's difficult to maintain such a narrow slit. And this is where Selectris energy filter comes in. With Selectris, the energy slit is so stable that even like within 24 hours, it just moves less than 1.5 EV. I run my microscope generally on a 72-hour runs, and I hardly even have to bother centering the slits. I never do this. All the data that you will see here, we only had to do the tuning at the start of the data collection, which you do for everything, and then we completely forget about the filter. On the cryos, it's stable, but also on the glaciers, and I'll show you some of the results from glaciers as well. So what happens is, because we are removing all this noise, your contrast is much better. So now this is a T20S sample. It's a proteasome 20S. It's a collaboration with Eugen Pritzow's group in Martin Street. Without filtering on the same grid, same setup, you hardly see any particles closer to focus. As soon as you do 10 eV filtering, all these particles starts to become visible. And as the defocus increases, your phase increases, and you can see that even there is a big increase in your contrast. And with increase in contrast, you also get a better signal to noise and hence higher resolution data from the same sample. So this is with 10 eV, we can get to 1.87 angstrom without filtering 2 angstroms. And also with number of particles, as I was saying before, that even by increasing, so now here I have 61% more particles with non-filtered data. Even there, my resolution only improves marginally because these B factors are coming in, you are hitting the wall. And I'll show you again one more example like this later. So we then tried this on apoferrite in our favorite sample to see what happens. And Holger gave a beautiful talk showing a wonderful atomic resolution apoferrite structure on day one. We also did similar, we obtained similar results with 360,000 particles, B factor of 32. So now here we are also using our CFAX, so CFAX Electris and Falcon 4. We can achieve 1.2 angstrom structure, just simply fantastic. And here is another movie that also I just look at the details that you see in this movie. You can see each and every individual atoms on all your amino acid side chains and tons of water molecules. But what we see is, in addition to this, we also see this wonderful densities for hydrogen atoms as well in this map. And this is a water molecule showing oxygen with this beautiful 2 hydrogen. And this is what we see this in the entire map. So yeah, we can achieve atomic resolution on a test sample. Then we also do something on a more challenging thing. So again, this is a GABA receptor here, which I showed you earlier. Same effect with filtering, contrast is higher and hence the signal to noise. And when we process this data, we push the resolution to 1.7 angstrom. And I think still now as well, this is one of the highest resolution structure for a membrane protein. Before that, it was all apoferritin or viruses, which could do such resolution. Now with a membrane protein as well, we achieved this in this case. And Garib Mashudov, who also gave a wonderful talk on day one, showing that how he is working with FMEC on building all the refining tools. He calculated this wonderful difference densities, where even at this resolution, you can start to see densities for hydrogen atoms as well in this map. Again, just as fantastic that you can see hydrogens in a membrane protein. All this work is published. There was a back-to-back paper with Holger and us in last year in November. It was a collaboration with George Shears and Takanori did all this data processing in this. So please feel free to have a read. But we wanted to see, can we do this onto more challenging asymmetric proteins as well? So this is another form of GABA. This is an asymmetric GABA, which I showed you earlier that we can obtain about 2.6 angstrom structures. 2 of this sample, same grids, we put it on a Selectris Falcon 4 system, and we can improve the resolution significantly. In fact, in this one case, Zopiclone, the data goes to 1.9 angstrom. And this is fantastic because Raju's team can now use all these structures to try and understand that how these drugs are modulating the receptors, and based on that come up with new, better drugs. So that was with the GABA receptor. We also wanted to see that what happens if you look at sub-100 kilodalton protein. So this is a streptavidin 52 kilodalton protein. It's a collaboration with Tokyo University. This is how particles look like. It's a filtered, it's a low pass filter to 20 angstrom in the ion. and we can produce 1.7 angstrom structure from such a small protein. And not only that, since we know that this is 1.7 angstrom, again, Garib's group at LMB Cambridge had a look at this map. Pietro worked on it, refined it, and again, in the difference density, they can start to see all the hydrogens and tons of water molecules. This is just fantastic because this is not apophratin. This is a sub 100 kilodalton, only a 50 kilodalton protein where we can see hydrogens. Then we wonder, so can we try this on other such challenging samples? So in this case, in collaboration with Steven Broham at UC Berkeley, we looked at this membrane protein. It's a 62 kilodalton ion channel from SARS-CoV-2. And again, with about 91,000 particles, it was about 3,000 images. We can push the resolution to 2.1 angstrom, and you can see all these details or numbers and numbers of water molecules and this channel, which can be used for the structure-based drug design to find a new therapeutics against it. And not only this, we worked on a series of small, flexible and challenging proteins, and we're still continuously working on these type of samples. And in every case, we are seeing that, yeah, because of this increased contrast, increased signal to noise, we are seeing much better, much higher resolution. So that was all about cryos. So just a few slides on seeing what happens with the glacier. So now this is the glacier, so 200 kV system with XFAX Electris and Falcon 4. Again, the proteasome 20S without any filtering, same grid. When we use 10 eV filtering, the contrast enhancement is just amazing. And with this contrast, we also get signal to noise. And signal to noise means improved resolution. So here, unfiltered goes to 2.34 angstroms, whereas with 10 EV filtering, we can get to 2.14. That's a 0.2 angstrom improvement in data. It's the same amount of data on both samples. And we can look at the B factors, and we can also see that, yes, filtering is helping, but also 10 EV is better than 20 EV. And to understand this in even better way, I drew the plots very similar to what Holger showed on day one. These are the linear plots. And what we can see is, indeed, that, of course, as the resolution is increasing, your number of particles go up, and at some stage you hit a wall. Now, if we just extrapolate a little here, and if you want to see that what would we need to reach 2 angstroms, then with the 10 EV slit, we will need about 370,000 particles of the same type to reach this to 2 angstroms. With the 20 EV, we would need about 1 million particles. And without filtering, we may not reach there. We just hit that wall where we don't have enough signal to achieve higher resolution. We also tried this on a range of different samples just to also convince us that this is true. On a GABA, again, unfiltered versus filtered, with 10 EV slit, we get 0.4 angstrom improvement compared to unfiltered data. We also tried asymmetric GPCR sample, a collaboration with Rado Sintanav and Patrick Saxton's group. This is an asymmetric membrane protein, very flexible. This is the same sample which they published last year, which went to 2.1 angstrom on the cryos. And now on the glaciers with Selectris, we can get to 2.6 angstrom. Now this is very good because just last year, even getting 2.6 angstrom on an apophratin was nice. And now we are getting this onto some membrane proteins like GPCRs, where this resolution you can already start to see all the water molecules and how all these drugs are acting. So we could even use glaciers for structure-based drug discovery in combination with Selectris. We have, again, the range of different structures, soluble proteins, membrane proteins, and every other week we are seeing more and more new structures coming out and all breaking all these new resolutions which were not seen before using the glaciers. So that was about glacier story. Now just last 3 slides showing you that we can also use Selectris because it's energy filter. We want to use it for tomography. So we started testing what happens when we do tomography with Selectris and Falcon 4.
Abhay, you have 5 more minutes, please.
Perfect, I'm almost done. So this is now with Jürgen Pritzko at Martin Street, filtered there on their microscope. Rubisco sample on a single particle, they can achieve 2 angstroms. And as of yesterday, they managed to push this to 1.8 angstroms. And the same sample with just 6,000 particles on tomograms, we can get to 3 angstroms. In another collaboration with Pei-Jun at Oxford and Diamond, with just 13 tomograms working on a very flexible HIV GEG virus-like particles, with 30,000 particles, we can get to 4.2 angstroms. Another similar project now with Florian Schor at IST Austria on EIAV GEG virus-like particles, again looking at this GEG formation. Only about 8 tomograms, 103,000 particles gets to 3.8 angstroms. And now we have collected more data and they have processed it, and the resolution have again improved significantly. And very soon you will hear about this story coming out. So that's all I wanted to show you on Selectris and Falcon 4. With this, also, I would want to thank all my collaborators who have been really fantastic, bringing me all these challenging projects to work with. And also, thank you, everyone, for listening to me. Thanks.
Abai, thank you very much for sharing your interesting and very nice results in electron density. So do we have any questions here? Misha.
Abe, hello, it's Misha Kudrashev. So, yeah, the question is, so you always show the comparison of non-filtered imaging and 10 EV slit, right? So it's the Selectris is very impressive, but so I wonder if there is any benefit of having a tight slit compared to something like 20 EV that people usually use on older energy
That's
filters.
indeed. So like we were also wondering for quite some time. And I have several experiments. I haven't showed everything here, but I don't know if you can see my slides still, but I showed you with this T20S that 20 EV, yeah, it helps compared to unfiltered data. However, with narrow slit, you can improve even further. Does that answer your question?
He says yes.
Yes.
So we also have a question from our chat. So can a normal Selectris filter help to get a similar resolution as an Selectris X?
That's a very good question. So we have 2 types of filter. There is Selectris and Selectris X. Selectris X is designed by keeping ultimate stability in mind. So we have like almost negligible distortions. There are hardly any geometric distortions. There are hardly any chromatic distortions. Now this is going to really help you to achieve high resolutions, especially in tomography, and also if you have large particles, because distortions are the biggest problem there. If you are working with small proteins, small particles, and you are not doing high resolution tomography, in that case, Selectris alone should be also fine. There we have higher distortions, very similar to the ones that we have on a K3 as well. So that's the difference between the two.
Okay, well, thank you very much for answering the questions. And thanks a lot for the lecture. So, and we'll have a quick pause before we continue with the next speaker.
Okay, thank you.
Okay, so let's move on to our next speaker. Bert, can you hear me? Oh, Bert, can you hear me?
Yes, I can hear you.
Oh, great. Okay, so it's my pleasure to introduce you our next speaker, Bert Pullman from the University of Groningen in Netherlands, who's going to tell us today about gating by ionic strength and safety check by cyclic DAMP in the ABC transporter OPUA. Bert, please, you have 30 minutes.
Okay, thank you very much. First of all, I would like to thank the organizers for a fantastic meeting and for inviting me. And what I will do today is to present the story of how we've used Cryo-EM to solve a series of important problems on how the cell regulates, controls its volume. And so more specifically, I will focus on the ATP binding cassette transporter OPUA. I will present the structural basis for cell volume regulation, but I will also focus on how the system actually senses ionic strength and is gated by ionic strength. And we also discovered recently that there is an additional level of control that involves the second messenger cyclic di-AMP. But before presenting these data, I would like to present a few aspects of the physical chemical conditions of cells, in particular bacterial cells. So what are the internal conditions? And what are the underlying homeostatic mechanisms that the cell uses to keep these internal conditions between certain threshold levels? And what I hope to show that is that this ATP binding cassette transporter OPUA is essential, but that under conditions it can also be a lethal protein. And so here's some parameters of E. coli. If we take the total metabolite concentration, then it is in the order of 200 millimolar, and almost half of it is formed by the amino acid glutamate. The major cation of the cell is potassium, like in most other cells. And both potassium and glutamate are important in the initial response of cells to, for instance, osmotic stress. Well, on top of that, cells can have variable amounts of compatible solutes from very low to submolar levels. And compatible solutes are small organic molecules with low toxicity that protect cells against osmotic and other stresses. And here I'm showing some examples, proline, the amino acid, and the other ones are actually quaternary ammonium compound that fulfill this role of being a compatible solute. Well, if we now go back to these internal conditions, then they lead in an exponentially growing E. coli cell to a turgor pressure, which is in the order of 3 atmospheres, which roughly corresponds to a difference in osmolyte concentration between in and outside of 120 millimolar. And these parameters are important to keep in mind. And also, these ions in the cell give rise to an internal ionic strength in E. coli in the order of 0.2 molar. But of course, these conditions, the conditions are not always the same. And when cells are osmotically challenged, you can have two scenarios, hypertonicity and hypotonicity. Under hypertonicity, the turgor pressure decreases and ultimately the cells can plasmolyze. And under hypotonicity, the turgor pressure increases, and this can actually lead to cell lysis. But it is not just the turgor that is changing under hypertonic stress, but for instance, also the ionic strength, which goes up, pH goes down, water activity goes down, the macromolecular crowding goes up, viscosity goes up, and so on. And these are just some parameters in the of the cytoplasm, but also at the level of the membrane, there can be changes depending on the osmotic stress conditions. And so what cells do, they try to adapt, and they adapt to hypertonic conditions by accumulating initially potassium and later on replacing the potassium by compatible solutes, and in that way restore the turgor. And under hypertonic stress, they rapidly try to get rid of these osmolytes in order to prevent the cells from lysing. And so they do so by using transporters, and for the exit of osmolytes, they use mechanosensitive channels. Well, today I will focus on the hypertonic stress and how cells accumulate compatible solutes, and they do so via this membrane protein, which is an ATP binding cassette transporter. And I will discuss the mechanism by which it is gated, but I will also show that it has additional control mechanisms to prevent overaccumulation. Because if you start with hypertonic stress, you need to accumulate compatible solutes, but if you overdo it, if you overaccumulate, the cell may lyse. And so the cell is essential, but if you don't control it well, the system is lethal for the cell. And so the two main questions that I try to address is, how do cells sense a decrease in cell volume upon hypertonic stress? And each of these parameters could in principle play a role. And also, how do cells prevent this excessive accumulation of osmolytes and thus prevent them from lysing? And so the focus is on this ATP binding cassette transporter, OPUA, which we study from lactic acid bacteria, but homologs are present in most, if not all, archaea and bacteria, and they are sometimes called BUS-A, GBUA, OPUC, OTA-A, PRO-U, and a couple of other ones. And what these systems do, they protect the cells under hypertonic stress by importing, for instance, glycine baiting. Well, here you see the cartoon of the architecture of this protein. It's a very complicated protein with many domains. So in blue, we see the substrate binding domain that capture the glycine baiting. In green, we see the transmembrane domain, which translocates the glycine baiting. And around it in yellow is some sort of a scaffolding domain that holds everything together. Then in orange, we have the nucleotide binding domain that powers the transport. And in red, we have the so-called cystathionine beta domain, which actually provides another level of control to this transporter. And as you can also see, this system is basically a homodimeric complex. So we've studied the system, we've purified the system and studied it in vesicles. And here I would like to emphasize something that we have developed recently. It's a vesicle system in which we co-reconstituted the transporter with another transporter that imports arginine and exports ornithine, and the conversion of arginine to ornithine is coupled to the synthesis of ATP, which can then be used by this transporter. And in this way, as I will show in a minute, we can supply the system with ATP for long periods of time. We've also equipped these vesicle systems with various types of sensors. Here we have pyridine for pH. Here we have a protein-based ionic strength sensor. Here we have a protein-based ADP sensor. And we have a few more that actually report on the status of these membrane vesicles when they are transporting glycine baiting. And here I'm showing the new system that we developed in the past few years with the classical system that most people are using, which includes ATP and an ATP regenerating system in the vesicles. And what you can see in such a system, the transport rate is high, but of course the transport levels already off after 15 to 20 minutes because of the decrease in ATP and the buildup of ADP. Well, in the newly developed system, we see that the transport rate is constant, at least for several hours, and we can actually let the system now run for up to days. And so we call this an open or out of equilibrium system because the system is continuously fed with substrate and products are excreted. And in that way, we can maintain a constant ATP-ADP ratio for long periods of time. Well, over the years, we've used both systems to study this ABC transporter. And here you can see an important piece of data which shows that the transport system increases in activity when the ionic strength is increased. So at around 0.2 molar ionic strength, which is basically the basal level of an E. coli cell, we see that the transporter is not active, but then if we increase the ionic strength, the system gets maximally active. Importantly, this ionic strength dependence of transport is highly dependent on the membrane lipid composition. And here I've plotted the transport activity as a function of anionic lipids, in this case, the fraction of the anionic lipid DOPG. And you can see here in the blue range, which is the physiological range of anionic lipids, we see that the system is inactive at low ionic strength, but fully active at high ionic strength. But for instance, if we have 20% of anionic lipids, we see that the activity is lower, but also that the system is no longer regulated. And so already several years ago, we postulated that the protein must have a domain that is sensing anionic strength, and in such a way that this domain interacts with the anionic surface of the membrane, in such a way that at low ionic strength, the system would be in the off state, whereas the electrostatic interactions between the membrane and the protein domain would be screened by high salt, and that would switch the system on. Whereas if we would have insufficient levels of anionic lipids, the system would basically work ionic strength independent. Well, in order to get structural information of the protein, we also reconstituted the protein into lipid nanodiscs. And in this case, the transport of glycine beta is coupled to the hydrolysis of ATP, which is stoichiometrically linked to the oxidation of NADH. And so we can see here that in the nanodisc, the ATPase activity is dependent of the concentration of glycine beta. We can see, like in the vesicles, that the ATPase activity is dependent of salt concentration, of ionic strength. And here we see the ATPase activity as a function of the ATP condition. So it looks as if also in the nanodisc, the system is fully functional, like we observe it in membrane vesicles, like we observe it in living cells. And so we obtained a total of 5 unique structures by Cryo-EM. I should say we've tried for more than a decade to get structural information from X-ray crystallography, and we have been relatively unsuccessful. We have obtained structural information from the individual domains, but not of the full complex. And so this changed when we started using Cryo-EM, and here you see 2 different structures. This is the apo inward-facing state of the protein, and you can see the membrane domain. You can see in yellow this scaffolding domain, and you can see the nucleotide binding domains. What we cannot see here are the substrate binding domain and this regulatory domain in red, because these domains are too flexible, at least under these conditions. Well, when we analyze the structure that was obtained in the presence of the substrate glycine beta and in the presence of magnesium ATP, we can now see the substrate binding domain, at least one of them, which is docked onto the membrane domain, and we obtain the protein in the occluded state. What you can also see is that the nucleotide binding domains, which are far apart here, have come close together, and they actually have the magnesium ATP bound under these conditions. And so what I will now do is guide you through different parts of the structure, and starting at the top here with the structure of the substrate binding domain. So we had already X-ray crystallography structures of the isolated domain in the open unliganded state and in the closed liganded state. And we see now in this Cryo-EM structure that we have a dock state, which has undergone a twist-like conformational change, such that the cation pi interactions between the tryptophan residues in the binding pocket and the substrate have been disrupted, and as a result, the the substrate glycine beta is no longer bound in this Cryo-EM structure, but has already been transferred to the transmembrane domain. And so overall, these three different structures nicely show how the protein from an open state goes to a closed state, and how it subsequently releases the substrate from the substrate binding protein and transfers it to the membrane domain. Well, if we now look at the top of the membrane domain, again for the protein in the apo inward-facing state and the occluded state, we see a series of highly conserved aromatic residues, which in the apo state point upwards, and in the occluded state take a different configuration. And we believe that this region actually plays an important role in the transfer of the substrate. And then if we look a little bit further into the membrane part, we see here that the apo inward-facing protein is open to the inside. And here we see now a cavity in the protein, which is surrounded again by hydrophobic residues. And we find some density, which is most likely corresponding to the substrate glycine beta. And that's why we refer to this as the occluded state. It's closed from the outside and it's closed from the inside. Let's then have a look at the scaffold domain. And so we knew already that a subset of ATP binding cassette transporters are longer, have additional sequences at the N-terminus, but we didn't know what they were doing. And it turns out that this extra piece of protein is forming a scaffold around the transmembrane domain, which you can see here in different side views and here in different top views. We've tried to delete this protein part because it's not present in all ABC transporters, but then the protein is no longer functional. And so the next thing that we did is we made a series of single cis mutants, and we labeled it with a FRET donor and then a FRET acceptor. And we determined the apparent FRET efficiency under a series of conditions: high salt, low salt, turnover conditions, inhibited state. But as you can appreciate from these histograms, the peak of this of these single molecule FRET histograms are always at the same position, indicating that this part of the structure is not moving, but sort of provides a rigid arm around the protein. We know from other Cryo-EM structures that actually the membrane part is actually highly dynamic, and most likely this scaffold is needed to hold the system together and allow a very efficient transport. So the scaffold domain was a novel feature that we discovered. We also discovered another new feature, and that is part of the nucleotide binding domain. Most of this nucleotide binding domain is not so different from that of other ABC proteins, but the regulated transporters, and I have listed here a number, they have an additional domain which forms an helix-turn-helix structure. You can see that the sequence of this region is not highly conserved, certainly not when compared to the rest of the protein. It's missing in non-regulated transporters, and it has several cationic residues on the one end of the helix and on the other end of the helix. And so this, of course, raised the intriguing possibility that this is in fact the ionic strength sensor. And so here I've positioned this helix-turn-helix motif with 3 of the cationic residues relative to the membrane surface. And this is actually the activated state of the protein. And here you can see that the cationic residues are indeed relatively close to where we expect that the anionic lipids will be. Well, to test that this region is involved in ionic strength sensing and gating of the transporter, We made a series of mutants. I will show one result over here. And so this is the transport activity of the wild type protein, now measured in living cells. And you can see that it is dependent of the internal ionic strength. And if we change these 3 cationic residues into alanine, we see that the transport has largely become independent of ionic strength. And we make similar findings if we mutate the additional positively charged residues on the other end of the helix-turn-helix motif. So far, I have not yet shown the red domain, which is the so-called cystathionine beta synthase domain. And what we discovered a few years ago, that this is actually a domain that binds cyclic di-AMP. It links those domains together, and it also fixes the nucleotide binding domains in a certain state. And here you can see a detail of that structure where we see the dinucleotide bound in this regulatory domain. And so you may wonder, what is cyclic di-AMP? It's a relatively new and recently discovered nucleotide. It's based off the nucleotide-based second messenger families, of which cyclic AMP was discovered first. And cyclic di-AMP was one of the last ones. Here you see the structure of the molecule, and it is formed in the cell by a dianylate cyclase from 2 molecules of ATP, and it is hydrolyzed by a phosphodiesterase. And so the activities of these enzymes are very important in microorganisms. So cyclic di-AMP is found in many bacteria and in archaea, not in all. For instance, E. coli doesn't have cyclic di-AMP, but it has cyclic di-GMP. And there are some organisms that also use a hybrid molecule, which is cyclic AMP-GMP. Mammalian cells do not synthesize it, but mammalian cells do sense cyclic di-AMP, and they use it to control the innate immune system, and they also use it to control the response against pathogenic bacteria. And so, important molecule on the one hand for the physiology in bacteria, and in eukaryotic cells for detecting microbes. And so here, back to our vesicle system, where we supply the transporter with ATP for long periods of time. And you can see here a high rate of transport over periods of hours. And if we add cyclic di-AMP on the inside, we see that the system is completely inhibited. What was a surprise when we analyzed the effect of cyclic di-AMP in nanodiscs, and what you can see in these two graphs, that in fact in the nanodisc, the ATPase activity is in fact stimulated by cyclic di-AMP, whereas the transport is completely blocked. And so how do we explain all of this? And this is summarized here in one of my last slides. So we have the transport cycle where glycine beta is bound by the substrate binding domain, which then delivers the substrate to the membrane domain. Upon the binding of ATP, the hydrolysis of ATP transfers the substrate to the inside, after which the release of inorganic phosphate in ADP resets the system back to the ground state. Then we have the ionic strength gating module that can keep the protein in the inactive state when the ionic strength is low, and activates it when the ionic strength is high. And this is, for instance, the conditions when a cell gets osmotically stressed. And so this is an on-off switching mechanism. And for many years, we thought that this was the only mechanism by which the system is controlled. But now through the structural analysis, we could show that, in fact, the cyclic di-AMP binding can provide an additional mechanism of control by locking the protein in an inactive state. It can still hydrolyze ATP because there is still some flexibility in the nucleotide binding domains, but this is no longer functionally coupled to the translocation of the substrate. And so we would say that this is an additional safety check that the transporter uses. because when cells get exposed to hypertonic stress, it starts to accumulate compatible solutes, which is sensed through changes in the internal ionic strength. But the internal ionic strength can vary under certain conditions, and so there is a risk of overaccumulation, and then this molecule, cyclic di-AMP, comes in and shuts the system off. And so this is also summarized here in words. Physical chemistry of the cell, I've focused on ionic strength, but also crowding and pH homeostasis play an important role in this overall process. And of course, each of these parameters is connected to how the cells regulate their volume. We've developed a new physical system for fuel. It's a versatile system for long-term production and use of ATP. And we've used it in many cases, not just to study this ATP binding cassette transporters. And then using Cryo-EM, we have elucidated several structures. We've discovered a new scaffolding domain. We've discovered a helix-turn-helix loop that plays a role in the ionic strength gating. And we've discovered how CBS domains play a role in additional levels of control of these transporters. I would like to say that CBS domains are probably the most widely used regulatory domains in biology. They are not only associated with this transporter, but with many other membrane proteins, including channels, but also associated with transcription factors, with enzymes. And in all these cases, they probably build one or another ligand to control the activity. In this case here, it is cyclic di-AMP that is doing the job. So this work, the structural work, was done by a very talented PhD student, Hendrik Sikkema, together with Jan Rijnberger in the group of Christina Paulino, with whom we had a fantastic collaboration over the past 3-4 years. Hendrik was also at the basis, together with Tjeerd, of developing this new out of equilibrium physical system for fuel. Marco van der Noort, another PhD student, did a lot of the functional assays, and Marijn de Boer did the single molecule thread studies. And several of the other people were involved in other aspects of the work, on biosensing, on cellular homeostasis, and so on. And here is a list of collaborators that we have on various aspects of this project as well. And this is a picture that was taken a few months before Corona started, and so we still had a retreat. And this was a retreat jointly of the groups of Albert Gustav, Christina Paulino, Dirk Slotboom, and my group. And these are 4 groups that are actively collaborating on many different projects. And with that, I would like to finish, and I'm more than happy to take questions. Thank you very much.
Thank you very much, Bert. Any questions? Let me see. So it doesn't look like we have any questions in the chat. Oh, yeah, there is a question. Albert, who's here physically.
Hi, Bert. It's Albert. So I know very well, of course, the story, and it's a fantastic story. But now I learned something new, and that's fantastic for me as well. So could you please answer how stable this cyclic di-AMP inside the cell, and how is it really sensed inside the human cells?
Yeah, that's a very good point. So what I'm showing here are 2 different enzymes. And what we are doing right now is to study how these enzymes are regulated. We know that their regulation is important because if the deadenylate cyclase is inactivated, which can also only be done under special condition, then actually the cells start to over-accumulate glycine 18 and the cells lyse. And so it's really needed to tightly control the enzymatic activities of these enzymes. In mammalian cells, there are known regulators of the immune system that have recently been discovered, as being the receptors that have cyclic di-AMP as a substrate. And actually, something that I also learned in the last year, that one of the main companies that develops the corona vaccines actually uses these cyclic dinucleotides as an adjuvant to actually help stimulating the immune system and getting us protected against the COVID-19 virus. Yeah, so there are a series of receptors that are also well studied. Important papers in Nature in the past year on this, but there is also still a lot to be learned on that, especially also on the regulation of the levels of cyclic di-AMP in the cell.
Okay, thanks a lot, Bert.
Thank you very much, Bert. Okay, we're moving to our next speaker. And we'll need a quick pause before. We will have a coffee break. 15 minutes, yes.
Leonid, good day. So I'm Misha Kudrashev. I guess it's time to start. Leonid, can you hear us? Oh, excellent. I see. So we have 5 very exciting talks for this afternoon. We will start with Leonid Sazonov from Institute of Science and Technology in Vienna, Austria. And Leonid, how's the connection?
Good, yes.
Okay.
Thank you.
Excellent. So Leonid will be talking about structures and mechanisms of membrane-embedded energy transduction molecular machines. Leonid, the stage is yours.
Okay. Can you see my screen? Yeah.
Not yet. Yeah, it's on.
You see my screen, yes?
Yes, yes.
Okay. Yes, so I'd like to thank organizers for opportunity to talk here. It's really fantastic conference. It's a pity I couldn't make it really in person. Hopefully next time. So I will talk about several of our projects on structures and mechanisms of membrane proteins which work as molecular machines. And as you know, energy metabolism, it happens at the membranes, either in bacterial membrane, shown here, or in eukaryotic cells in mitochondrial inner membranes. And most of our proteins come from these 2 membranes. And so, long time ago, in about 1997, when I started kind of structural work, I chose to work on complex 1 because at that time it was really a black box. There was no structures whatsoever. Even so, the structures of other complexes of respiratory chain were known mostly, like complex 3 or 4, or most of ATPase. But complex 1 was really a black box, in part because it's the largest component of the chain, up to 1 million Daltons and 45 different subunits. So of course it's difficult to crystallize. And also, like my general life principle, I'm interested in unknown molecular machines, proteins which work by some unusual ways, which we don't know yet. But when you discover them, they're really, exciting and amazing. So that's what kind of motivates me. And so, I will talk today about our work on complex 1, but also on the transhydrogenase here, which is kind of forgotten member of respiratory chain, and also work on ATP synthase, more recent. And if we talk about mechanisms, almost all proteins in the chain work by different mechanisms. For example, complex 3 has unique Q cycle. Complex 4 works by kind of direct chemical coupling of reactions. ATP synthase, of course, conformational machine. And complex 1, as we thought originally, was really conformational machine, but now our mechanism is a combination of conformational and electrostatic coupling. And the same applies to transhydrogenase as well. So I think there are some common kind of design principles of this non-directly coupled redox proton pumps, which I'll discuss in the end. So first structure we solved was of the bacterial complex 1. And that was done by X-ray crystallography. And it was at the time largest asymmetric membrane protein solved by X-ray. So that was a huge effort. Took us like 10 years to optimize crystals and solve the structure. Because most of it was de novo, not known folds, 16 different subunits, 9 iron sulfur clusters. So it was a challenge, but it also formed a basis for the core of later mitochondrial complex 1 models, which is much larger. And also 2013, when we published it, was kind of start of new era of Cryo-EM. And we also adopted it. So at the time, what we thought about the mechanism of complex 1 was that it's conformationally coupled, so that redox reactions happening around Q site. So this is electron acceptor of electrons coming from NADH via iron sulfur clusters towards quinone binding site at the interface between peripheral arm and the membrane arm of complex 1. So this creates temporarily charge in this area, like double negative charge. It's 2 electrons, so it's on a microscopic scale, it's a huge driving force. And we thought that it drives kind of in a kind of reminding of coupling rod in a steam engine through the central axis of the membrane domain, which has many broken helices, so it's flexible. And it also has many charged conserved residues on it, and also many waters, as we know now. So it's flexible and conserved and charged. So we thought that would be kind of transition between 2 states shown here on top and bottom, which results in opening and closing of channels and total translocation of 4 protons across the membrane. But now it's changed, our proposal, but I will describe it later. So when we switched to Cryo-EM, the first structure we solved was of entire mammalian complex 1 using ovine enzyme. And this was the first complete structure because the other structure from Cambridge was about half complete at the time. And it took us about 2 years to solve it because it was not yet fully advanced technology as we have now. So now I think we could solve like new structure of complex 1 of new species in about 2 months. So that's really amazing if you think about it. If we still would have to spend years and years on crystallizing these huge proteins now, I think our progress would be much, much slower. I think main bottleneck now is really to analyze new structures and write the papers rather than grow crystals. So it's quite amazing time. And so the mammalian complex 1 has 30 extra subunits in addition to the core subunits conserved from bacteria. But the core subunit structure is really the same, more or less, shown here in color. And all the extra subunits provide a shell around it in gray. So most of them are, we think, stabilizing, like, for example, this large area of rigid membrane, rigid helices on this side of the complex. But some of subunits might be also providing additional regulation of the activity by links to like redox state of the mitochondria or to fatty acid synthesis and so on. But the core is the same, so the mechanism should be the same throughout the species, we think. So to understand complex one, it's important to look at evolutionary origins of the complex, and it comes from the combination of the MRP-like complex. So it's MRP antipolar converts or couples translocations of sodium or potassium to protons, and its subunits or similar subunits are now found in many redox-driven proton pumps, like complex one. So antipolar-like subunits here are shown in color, green, yellow, and orange in this case. So there are 3 similar subunits in complex one. And of course, we think each of them is responsible for one of pumped protons, and the additional one is linked to the Q reactions and E channel here. And the original ancestor was probably membrane-bound hydrogenase, as shown here, which contains either 1 or 2 antipolar-like subunits. They're also found in the chloroplast, complex 1, NDH. So we think it's important to know the structure of this ancestor. So Julia Steiner in my group undertook this challenge to solve structure of the MRP complex. In this case, from thermophilic bacteria, Flaviothermus, and it's 7 subunits. So green and yellow are antipolar-like, and the others are similar mostly to small subunits of complex 1. But with this complex, we encountered probably the most severe case of kind of bad behavior of protein on a cryo-EM grids. So in ice, as usual, it formed this aggregate stacks in whatever conditions we tried. And so Julia tried to optimize this in huge heroic amount of work using different detergents, additives, different constructs, amphipoles, regulated grids. Nothing helps. So in ice, it was always aggregated. But then we moved on to continuous support, like carbon, which we use, and all the problems vanished. And I think it's mainly because on carbon, protein is attached to the carbon and it avoids air-water interface, which was mentioned before. It's really damaging to the protein. And so when we moved to carbon, we got nice spread of particles on it. And now we use it almost for all our projects. So why? Well, there could be some problems if preferential orientation, but it's easily solved by collection at tilt. So it's no problem really. People are worried about high background, but we found if you use homemade carbon layer about 1 nanometer, it's really not a problem. It does not diminish resolution in any way, at least for proteins 200 kilodalton or more, which we tried so far. But advantages are many. So protein avoids air-water interface, denaturation and aggregation is prevented. You get even spread of particles because they all see only the same surface. There is no split on, like most particles often go just to carbon. And you need like 20 times or more lower protein concentration, which is often important. And you avoid problems for membrane proteins because usually detergents co-concentrates with protein, but here you don't need high concentrations. And it's also much easier to handle and more reliable than graphene supports. So we think it's kind of silver bullet, which Bridget mentioned earlier, at least for membrane proteins. And so it was reviewed in our science paper recently. So using this, we could solve the MRP complex structure at 2.9 angstrom resolution. And it's obligatory dimer of 7 subunits in a monomer. And I don't have time to discuss details, but it suggested to us that the mechanism is probably conserved from complex 1 because you have 2 antipoters and small subunits hold the site for the sodium. In this case, in complex 1, this would be quinone charge intermediates and they electrostatically interact with antipoters driving proton translocation. So coming back to respiratory chain now, proteins exist usually not as individual complexes in a chain, but as a super complexes, either 1 + 3 + 4 respirosome or 1 + 3. ATPase is usually a dimer. Also, there is complex 3 and 4. And so we study these super complexes as well. The function is not entirely clear, but it could be stabilization of individual complexes. A protein packing is improved, so you could put more in the same area. And also we suggested that, and others previously experimentally, that such architecture may reduce reactive oxygen species production, which are harmful, of course. And we propose it's because electron flow is kind of streamlined in this case, and you have shorter living radical intermediates, which would prevent excessive Ross production. And so we still work on this and other supercomplexes. And also on ATP synthesis or ATPases. So one of the first ones we analyzed was a VA type ATPase. So it's related to the V ATPases, which use ATP to pump protons, in contrast to F-type ATPases, which use proton gradient to produce ATP. And VA from archaea or some bacteria is in architecture, they're close to V-type because they have this extra color and extra peripheral stalks. But in function, they could be either pumping or consuming protons depending on species. And there was no structure of A-type ATPase before, so we solved it from Thermophilus in the 3 main rotational states and 2 substates of those, which allowed us to put together this movie, how it works. And I don't have time, but just one main message from this was that really you need very substantial conformational plasticity in a connection between V1 and V0 domains. Because as you can see, F1 wobbles quite a lot relative to VO. And this extra stalks might help in this case if the main direction is from VO to V0. So it's other way around to F ATPases, which may require higher plasticity. And of course, arguably the most important ATP synthesis is the mammalian ones, as is human. And surprisingly, despite many structures of ATPases recently, till last year, there was no complete structure of mammalian ATP synthesis. So we solved it with Gergely-Pinke here. So this was the first complete structure and published last year. At the same time, John Walker group also published essentially the same but bovine structure. So one interesting aspect of this was that we were interested in a role of ATP synthase in a permeability transition for PTP. So this is very hot and controversial area at the moment. Some people believe that ATP is synthase is involved. Some are not, some don't believe it. So we wanted to look at this in a more detail. And one surprising feature of the structure is that inside the C ring, it contains two types of lipids. One kind of on top, close to F1, apparently rotates together with the ring, because in an averaged map of all states, it disappears, but it's present in a single state. But the other lipid on the other side appears to be fixed to peripheral stock somehow, because apparently it does not rotate with the ring, as you can see here. And so what we proposed was that this could be a key to the PTP transition, because it happens when calcium is present instead of magnesium. So calcium binds here in F1 sites, and calcium is of course bigger in diameter than magnesium. So we think it would introduce some additional conformational changes in V1, which would propagate through peripheral stock to this area here, which is kind of hotspot for several mutations which are known to interfere with PTP. And this area is connected through the E subunit to this lipid, which is fixed inside the C ring. So what we propose is that when you have calcium and conformation changes, kind of pull out this plug, lipid plug out of the C ring. So it's getting distorted by incoming waters. And so the other plug also comes out and you get the opening of the pore. And we've seen some kind of intermediates resembling this when we apply calcium. And of course, we're trying to solve higher resolution structures of these states. So coming to the more recent work on mechanisms, Domain Compute, really excellent PhD student in a group, solved structure of like two major problems. So first he solved structure of the transhydrogenase, which works as using protons, one proton per cycle from the respiratory chain to reduce NADP to NADPH mainly. And this is used to detoxify reactive oxygen species. So it's important metabolic enzyme, and structure of the entire enzyme was not known. And so we solved it to 2.9 angstrom resolution. It's a dimer of two subunits, so you see here. And it has a membrane domain, NADP binding domain in red, and NAD binding domain in yellow here. And I think it should, yeah. So in this movie shows that it's really amazing degree of conformational changes in this enzyme. You see entire NADP binding domain swivels about 180 degrees during catalytic cycle. And this happens in both monomers during the cycle like this. It's really, I think, amazing. And the reason for this is when hydrid transfer for NADP reduction happens between two domains in a top position, it then swivels to the bottom position and electrostatically interacts with the histidine in a proton channel. changing its pK, and this results when domains swivels back in proton translocation. And so this is consistent with many mutations and so on. And finally, my timer is difficult to see. Okay, I think still a bit of time. So Domen also was instrumental in our, now we think we more or less really solved the mechanism of complex 1. And this was possible because he saw many structures of ova in complex 1 in many different redox conditions and high resolutions up to 2.3, 2.2 angstroms. So it's resolved lots of waters shown here in the red spheres. You can see many in the hydrophilic arm, of course, but also many in the membrane arm, as Werner mentioned before. And the mechanism is really related to two main conformations of complex 1, which we see it's open and closed. We see transition between them here. And this is coupled to huge conformational changes at the interface between two domains. You see entire helix rotates here, and also many loops and helices shift and change conformation at the around Q binding site. And due to high resolution, we could see many lipids, so the substrates, cofactors, ions, post-translational modifications, never seen before, but known from biochemistry. And of course, water molecules, which are essential to understand the mechanism because they are kind of stepping stones on a proton transfer by Grotthuss mechanism. And you see many of them in the peripheral arm, but not between the clusters. So it's in contrast to some predictions. And also many in what we call central axis of the membrane domain, which I mentioned before, around charged residues in the middle of the membrane. And here I just want to mention briefly that we developed refinement script because, of course, in Cryo-EM carboxylates density disappears often. And so if you refine it as it is, in real space, it will be pulled into backbone. So we have a script which first allows to refine B factors of this such into high values. And so later they don't interfere with refinement. We find it really important. It's described in our molecular cell paper. And so in complex 1, we have several key loops around the Q cavity in the interface. And so they are related to kind of surround from all sides quinone head group when it's bound in a cavity. And if you look at the closed state of the complex, quinone is really very tightly engulfed in a complex. There is no space for anything else. And a head group, around head group, there is no space for waters either. But in an open state, you can see cavity is expanded quite a lot. So this allows for quinol to come out or for the quinone to come in. And also this opens a channel to the cytosol or to the matrix space on the other side. But in this state, within this channel does not produce any chemical protons. It's not involved in chemical reactions, in contrast to some other suggestions. And when complex is reduced, these loops kind of push quinol out of the cavity. And so you can see these movements in the movie here. There's a key loops and there's a kinon here. And in a closed state, it's bound deep in a cavity, and then it's pushed out of the cavity, or other way around, of course.
Leonid, we're running short on time.
Yeah, okay, I'm finishing. So in proton pumping mechanism, we see waters all across the central axis. We see access across entire distal and deep 5 subunits. Here also from the another neighboring antipota subunit, but all the other pathways seem to be closed as far as we can see from waters. But there is only one empty area in an open state between the Q cavity and the rest, which you see it here. There is no waters, no access. But when this key helix, which rotates, as I mentioned, when it rotates, it's free up space for waters, which establish this connection. And so in the mechanism, in an open state, we propose that this connection is blocked, and we have protonated residues in this area called E channel. And in a closed state, when quinon comes in, it accepts electrons. It has to be protonated, but all the pathways are closed, so it can be protonated only through the central axis. And so this helix rotates, side axis opens, and protons come in from the central axis. And so this leads to electrostatic, brings a charge to double negative charge close to antipoders and initiates electrostatic interactions leading to proton pumping eventually out of the distal subunit. So in this mechanism, we have combination of conformational changes in the Q area, which brings the charge action close to antipoders, and then this leads to pumping out. And to answer your, Misha, question earlier, why it has only this distal output, we think this is because this way protein can fully control electrostatic interactions and proton transfers through the central axis from the Q to the very end. Because this allows it not to have any conformational coupling in antipoders, which we don't see. They really don't change the conformation even under turnover. So this is mechanism which is unexpected combination of conformational and electrostatic interactions. So from transhydrogenase and complex 1 work, we think there are some common principles of such pumps, that redox reactions lead to conformational changes and place charges near proton transfer residues, and then the rest is done by electrostatic interactions. So I want to thank key people involved in the work I described and other group members. Thank you.
Mm-hmm. Thank you, Leonid. That was an incredible tour de force on what will probably become the textbook knowledge very, very soon. We have time. We're a little behind time, but I think we can have a question or two, if anyone has it. Arthur?
Hi, Leonid. It's Albert. So fantastic work, of course. So I have a very quick question about the lipid plug you mentioned. So is there any specificity in the lipid, or it can be just any random lipid to serve as a plug?
No, we think it's really quite unusual lipid because it has only one hydrophobic tail from density, in contrast to most lipids, which have, of course, two. And it needs to have only one because otherwise it would not fit in this area. So this is some form of lysolipid, which of course exists in mitochondria, but exactly which one we don't know yet. Yeah.
Okay, thanks.
We have a question from the chat. So how is it possible to rule out that the open state of the ND5 connected waters is a transient state? Thus, the structure on its own does not rule out that the protons can pass through other antiporters.
No, I mean, we have both open and closed states at high resolutions, and we see waters in both states. And there is clearly no possible connection from the other antipoters into the periplasm or intermembrane space. There's really no way, because it's large hydrophobic residues. And yeah, as I say, there is no difference between different states. But there are, of course, huge differences in the rest of the structure, except for antipoters. So I think we can really exclude possibility of not observed yet conformational changes, which would really have to be huge to achieve any connectivity from antipoters, from other antipoters into the periplasm. And also sequence comparisons, if you look at them, it's clear that ND5 subunits is very different from the rest, especially in this connection area to the periplasm.
Okay, a quick follow-up question, if there are any other transient states?
Yeah, I mean, we've seen that if you have only NADH and no quinone, there is one specific conformation, which I mentioned, in which one of the key loops is embedded further into the Q cavity. So this would help to eject quinol. And otherwise, there are no major changes which we could see, and we studied really, I think, all possible conformations.
Mm-hmm. Thank you, Leonid. In the interest of time, we will have to thank you with a live applause, hopefully. Thank you for an amazing talk, and then we will have the next speaker shortly. Hello, Inga, can you hear us?
Yes, I can hear you.
Great. So it's our pleasure to have Inga Henold. She is from University of Frankfurt, and I'm very proud to be her colleague from across the street. Inga will be talking about structural basis for controlled potassium uptake by KUP involving transinhibition and cyclic DIMP binding. Inga, please go ahead. We're a little bit delayed, so time is yours.
Okay, thanks a lot, Misha, and of course, thanks to the organizers and particularly to Albert for inviting me and for putting together this exciting meeting. So it's been a great pleasure thus far. I slightly changed my title as I will talk a bit more broadly about potassium transporters and channel in material survival in general. And I was fortunate that Bert already introduced a bit to you how important potassium is to bacterial cells, so it can be quick on this slide. He already mentioned how important it is in osmoadaptation, both in the environment and also when invading a host system. But potassium also is very important when surviving on dryness, for example, or when communicating within a biofilm. And a generally rather high potassium concentration is required also for the activity of various enzymes like the ribosome, for example. And as such, it is important to notice that the potassium concentration within a bacterial cell usually are quite a bit higher than in eukaryotic cells. So there's concentration ranging from 100 to 400 millimolar usually. And another thing I would like to mention is that also the membrane potential is often a little bit higher, and it is built up by protons, which will become important throughout my talk. Well, being that important for the survival of bacteria, it doesn't come as a big surprise to you that there are various potassium uptake and release systems in bacteria. And those probably most of you are familiar with are KCSA, MTHK, or GSUK, which structurally have been studied in great detail. However, I'm sorry, I have to tell you that we have no idea of what these channels actually contribute to the survival of bacteria. So this is one of the mysteries to be solved. Then there's several potassium efflux or release systems. They have mentioned the mechanosensitive channels, which allow the rapid release of potassium and also other osmolytes. And then there's active potassium transporters, which mostly are used to establish or to adjust the internal pH. And then this is what I would like to focus on today. There's three classes of potassium uptake systems. There's the major player KTRA-B and TRKAH, which are potassium channels and still the major player. And this is why I mentioned the membrane potential, because due to the high membrane potential and since it is built up by protons, this channel is able to accumulate a high concentration of potassium inside the cell. The second one is an active potassium pump, KDPF-ABC, and the third one I will talk about is the potassium proton symporter family, KUP and KIM-A. Now we'll start to introduce to you KTRA-B, which is one of the two major players for potassium uptake, active at neutral to alkaline pH, and can serve the uptake of potassium at rather high external potassium concentration of down to 50 micromolar. We've been studying this for quite a while, and the work I'm going to present you here has been done in collaboration with Janet Fong from the MPI of Biophysics, with whom we collaborate a lot on the structural approach that we do in the lab. So just to keep that in mind. So KTRA-B is a potassium channel, as I already mentioned, but it's quite a bit different from the 2TM potassium channels, as it forms a dimer within the membrane where you have two parallel pores, and you can better see this in this representation here. And so each protomer has 4 fused 2TM transmembrane segments forming a single pore, and then we have 2 parallel pores, which are actually regulated by 1 octameric gating ring that associates to the cytoplasmic site. And this regulatory ring is known to bind nucleotides by which it undergoes conformation changes. By functional studies, we could show that just the channel subunit alone, so just the transmembrane domain alone, is sufficient for the uptake of potassium, both in the presence and the absence of sodium. While if we then study the complete KTRA-B system, we see that it is dependent on sodium, on ATP, and recently it was also shown that it is regulated by the binding of cyclic AMP, which inactivates the channel. So there are 3 mechanisms of regulation: sodium, nucleotide binding, or ATP, ADP binding to the central part of the ring, and cyclic AMP binding to these C loops here. We've been studying various aspects here, but the only thing I would like to touch upon today is how ligands regulate channel gating and the insights that we have at the moment. So the first structure that I showed you here was solved by the group of João Moraes Cabral in the presence of ATP. And you see these helical hairpins here, which are broken in the ATP bound conformation. What we managed to achieve structures of together with Janet was of the ADP bound conformation, where you can already see quite a conformational change because these helical hairpins now are gone, but instead the extended helices reach into the octameric ring, forming a direct contact with the octameric ring and with the ADP binding site. So by just running a morph of our structure and the structure solved by Joao, you can appreciate the conformational changes, and based on the conformational changes you see, you can also guess what is the effect of these conformational changes. So what we have is we have a conformational change of the octameric ring from a 4-fold to a 2-fold symmetric conformation, which then changes the conformation of the extended helices, and which in consequence free or seem to free the intramembrane of ADP. This gate is fixed in a distinct conformation where we see only one distance distribution when spin labeling the intramembrane loops, while in the presence of ATP, we get a broad distance distribution arguing for the flexibility of this loop. So what we think at the moment, how this system is regulated is that by the binding of ATP and by sodium, which we don't quite understand at the moment, and possibly by other factors, we have a conformational change in the octameric ring, which triggers a conformational change in this extended helix for forming this helical hairpins here, which then regulates the flux of potassium. And as said, we might have additional regulators like the binding of cyclic AMP, which play an important role here. The second system I would like to introduce to you is CUP and KIM-A, which is the system that comes into play if we have acidic pH. So KTRA-B is only active at neutral to alkaline pH, while KIM-A particularly is active at acidic pH, while it has similar affinities to KTR and TLKA. But instead, or other than KTRA-B, it's not a potassium channel, but it's a secondary active transporter which couples the uptake of potassium to the uptake of protons belonging to the APC superfamily. Of course, we also would have liked to have further insights here, and that's why we started studying this. And at first, we actually confirmed the hypothesis that this system is a potassium proton symporter, as we saw a strict dependency on the pH on how much potassium is taken up, while the addition of CCCP, which abolished the PMF, also abolished the uptake of potassium. And when reconstituting KIM-A into liposomes, we vice versa saw a dependence of the proton uptake on potassium, which then resulted in the ACMA quenching, as we have shown here. Of course, we were eager to know how this, or what is the structural basis for the proton-coupled potassium transport. And for this, again, with Janet, we teamed up solving the structure of the KIM-A uptake system. So you see the structure here and the fold here. So we have 12 transmembrane helices and the cytosolic domain, where the cytosolic domain is swapped with respect to the transmembrane domain in the dimer that it forms in the membrane. When looking into this in detail, then you can see that the first 10 helices show 2 inverted repeats with broken transmembrane helices in transmembrane helice 1 and 6, you can see here, which is well, probably quite familiar to many of you, as it is the typical Lu-T fold known from many secondary active transporters, where the substrate binding site is expected to be at the location of these broken helices here. Transmembrane helices 11 and 12 then connect to the cytosolic domain, and the cytosolic domain itself is formed by a parallel 5-stranded beta sheet flanked by 4 helices, which then extends to 10 beta sheets in the dimer. Interestingly, the dimeric cytoplasmic domain has a similar fold to phosphopantothenium adenylyl transferases, which already when solving this structure led to the hypothesis that there might be regulatory nucleotide binding, and I will come to that in a minute. Important for the dimer formation, that's also quite striking, I find, is only this, unstructured loop here, which forms a connection to the cytosolic domain of the other protomer. We then had a further look at a closer look at our structure, and what we find is that the structure we solved is actually an inward occluded structure. So you can see that it is quite restricted from the outside, while we have a tunnel reaching from the cytosol into the cavity or into the binding site. However, this is still restricted by some side chain interactions. When we looked in closer detail, we also could identify non-protein densities, 3 in total. 1 at the location where we expected substrate binding, namely at the broken helices, and 2 at the tunnel below. And of course, our next aim was to solve or to answer the question, what's the role of these 3 potassium ions, whether these are all substrate or which residues are important for substrate binding. For answering this question, we make use of a very simple functional assay, namely a growth assay, where we determined the growth rates at different potassium concentration and plotted them against the potassium concentration with the Monod equation. And you can see that for the wild type, we have an apparent affinity for potassium of 0.09 millimolar. We then at first mutated the residues around this potassium ion at the expected substrate binding site, and you can see that particularly aspartate 36 and tyrosine 43 led to complete abolishment of potassium uptake or growth, while mutating the other residues surrounding this potassium ion led to a reduced apparent affinity, which is actually what you would expect and confirmed our hypothesis that this is the substrate binding site. We did the same approach for the other residue that was involved in binding the 2 potassium ions in the tunnel, but actually when mutating this residue, so the aspartate here, we saw no effect. So this was to some extent surprising, as we expected to see something, but led to the hypothesis that there must be another role for these 2 potassium ions. And then there was another striking amino acid, also highly conserved, which is this glutamate here, which is too far away for the coordination of the potassium ion. But upon mutating this, it also led to the abolishment of growth, which led us to the hypothesis that this residue might be involved in proton coupling. Of course, we also wanted to test this hypothesis, and the first we did a counterflow assay in which we loaded our cells with high potassium concentration and then diluted them to high rubidium concentrations, while at the same time we abolished by the use of CCCP the protomotor force. And the hypothesis would be that if the residue, the glutamate residue is important for the proton binding, the counterflow that we would measure here would still work, while we do not see active transport. And this is what you see for the wild type. So you see the nice exchange of the potassium inside the cells against the rubidium, because you see a decrease of the internal potassium concentration. And we see a quite similar result for the glutamate to alanine mutant. So this residue is still able to, or this mutant is still able to exchange rubidium for potassium, which is in line with our hypothesis. While our control, the aspartate 36 to alanine mutant, did not exchange any rubidium for potassium, as it can no longer bind potassium. So this was in agreement with our hypothesis that the glutamate 233 is in fact the residue that binds the proton and couples the proton, the potassium transport to proton transport. The other hypothesis was that the other 2 potassium ions might be involved in a transinhibition mechanism. And the way to test for this is to look at growth at increased potassium concentration. Because if you would not have this potassium, this transinhibition mechanism, cells may accumulate toxic concentrations of potassium at very high external potassium concentration. And this is exactly what you can see here, because when we grew the cells expressing the KIM-A mutant with the alanine instead of the aspartate, you can see that there's hardly any growth at 150 millimolar, which confirms our transinhibition mechanism, and which again is in agreement with what Bert has told you 2 talks ago, where they also expect some inhibition mechanism avoiding accumulation of too high concentrations of the compatible solute in this case. Well, but I already told you that also for the cytosolic domain, we had the idea that there might be nucleotide binding, and this came not from nothing, but there were data already that cyclic AMP also plays an important role in regulating potassium uptake system. And it was known that the gene expression is regulated by cyclic AMP, and other potassium uptake systems like KTA were already known to be regulated by cyclic AMP. So we tested whether also KIM-A is regulated by it, again, by a growth experiment, by the co-expression of KIM-A with the cyclase that produces cyclic AMP. And when doing so this is the triangles, in fact, we see no growth of the cells at low potassium concentration where we would require the uptake systems, while upon the expression of the inactive cyclase, we see normal growth. So this was the first confirmation that there is a dependency. So we were, of course, eager to get the structure of this. And this is what we achieved very recently. So you can see now, again, a structure now solved in amphipoles, where we see very similar features. We also see the potassium binding, but we also see the cyclic diAMP bound to the cytosolic domain. And interestingly, this cyclic diAMP is coordinated mostly by side chains within the cytosolic domain, but there's one arginine that originates from helix number 8, that also is important for forming salt bridges with the cyclic diAMP and for its coordination. And thus our hypothesis, of course, at the moment is that this is the residue important for the inhibition. For testing this, we again used growth assays where we express KIM-A and KIM-A mutant, where the arginine is mutated together with the active or inactive cyclase. And we can, I guess, appreciate that in fact, when mutating the arginine, the co-expression with the cyclase no longer inhibits the protein, while at the same time, we could show that the cyclic diAMP can still bound. So we, by now, conclude or suggest that the transport mechanism of PUP is an alternating access mechanism where either side can be exposed to bind potassium, first from the outside, potassium and proton would bind. Then upon a reorientation, there would still be inward occluded state until the proton is released, and then also the potassium ion can be released. The structures that we solve now both represent the same inward occluded inhibited state, where both the trans inhibition and the binding of cyclic diAMP add up for the efficient inhibition of the complex. The last protein I would like to talk about is the KDP-FABC system, which is the rescue system at potassium limitation, as it has a very high affinity for potassium. The system is quite unique as it forms a complex of 4 subunits. It contains the P-type ATPase, the smallest known P-type ATPase, which is responsible for ATP binding and hydrolysis, so binding via the N domain and then phosphorylation of the P domain, which then couples this to the uptake of potassium. The coupling relates to the second domain, KDPA, which might look familiar to you because it is like KTA-B, the potassium channel-like subunit. So it belongs to the same superfamily, also has a selectivity filter very similar to KTA-B and an intramembrane loop. So one of the key questions here is how does KDP-FABC couple the uptake via a potassium channel with the ATP hydrolysis in the P-type ATPase? There are 2 further supplements, KDPF, which has a lipid light stabilizer function, so it can be basically replaced by adding further lipids upon purification. And there's KDPC, which is a paraplasmatically oriented protein, which is yet of unknown function. Yeah, and as already mentioned, together, this complex actively transport potassium. Importantly, it's strictly coupled. So you can see here the ATPase activity. You have only high ATPase activity if you add high concentrations of potassium, while you only have the transport of potassium shown in this transport assay here upon the addition of ATP. And the key question is, how is that possible? Based on the first structure that was solved, a coupling helix translocation mechanism was suggested in which potassium ions would bind to the selectivity filter. This would then, via a Grotthus mechanism, via this water-filled tunnel that was identified in the first structure, would protonate here the canonical binding site, which then would trigger ATP hydrolysis. And the ATP hydrolysis would lead to a conformational change, which via a coupling helix, that's like the coupling helix mechanism, would pull open the intramembrane loop and release the potassium ion to the cytosol, while at the same time, the KDPC domain might serve as an external gate. Well, at the same time, we were also working on this, and when we, together with Christina Paulino from the University of Groningen, managed to solve structures of both the E1 outward-facing and E2 potentially inward-facing state, we initially were quite frustrated when we saw that there were no such conformational changes. So a superposition of the transmembrane domain of the channel subunits showed no changes, and also the KDPC domain showed no conformational changes at all. However, a further inspection of these two states then revealed that again we have this intersubunit tunnel, which in the E1 state runs all the way from the selectivity filter to the canonical binding site, while in the E2 state it is blocked at the interface between the channel subunit and the p-type HPase, while at the same time a second half channel opens from the canonical binding site to the cytosol. And further, we observed additional non-protein densities within this intersubunit tunnel, such that we suggested an alternative transport mechanism via this intersubunit tunnel. So we suggest that the ions bind to the selectivity filter, move all the way through this tunnel to the canonical binding site. At the canonical binding site, ATP hydrolysis leads to a conformational change such that the highly conserved arginine lysine residue pushes the ion off the canonical binding site into the cytosol, and then the second half cycle leads to the start point again. Well, this was a nice model, we thought, but of course there was quite a discussion because assigning potassium ions is difficult, and the question is how would the ions be forwarded through this tunnel. So we moved on, and this is quite new data that I would like to present to you now, and aimed at solving higher resolution structures, possibly reaching a better assignment of the extra densities. And the higher resolution structure that we could solve recently is a potassium-loaded E1 ATP structure. So it's inhibited in the E1 state by the use of AMP-PCP, which you can nicely see here, wedged in between the P domain and the N domain. The structure had such high resolution that we can also identify two cardiolipin molecules that we show by mutational studies are important for the stability of the complex. And we could nicely, again, modulate or assign this tunnel that runs all through the protein from the selectivity filter in the channel subunit to the canonical binding site in the p-type ATPase. What you can also nicely see by this profile here is that there are two restriction sites, one at the selectivity filter and one at the conserved phenylalanine residue here, which restricts the tunnel and seem to be important checkpoints to which I will come in a minute. When looking at the structures further, we also could see a lot of additional densities now, since we had higher resolution, and there was one reason, and since we used higher potassium concentrations, we believe. But we also we really wanted to make sure that we can nicely assign the densities this time. So that's why we also solved the second structure. And in the second structure, we loaded rubidium ions. And we used a little trick for that, because there is a mutant that is able to transport rubidium ions, which is mutated in the selectivity filter with this mutation. And the idea behind that was that due to the higher atomic number of rubidium ions over potassium ions, we would reach a higher scatter. And in fact, when we compare now our two structures that we reached, we can see a twofold higher scatter of the potassium ions, both in the canonical binding site and in the selectivity filter, which are quite in favor of our transport mechanisms. So being now very confident that this is really the transport pathway, we moved on and teamed up with Robin Corey and Phil Stansfield at Oxford University, and we ran MD simulation. In the first MD simulation, we ran a short simulation where we let the ions within the tunnel relax, and immediately we could see that in the simulation, the ions take up different position. One here, just before the canonical binding site, and another one to which I will come in a minute. We then looked at the APBS energy profile, and you can see that it is quite in favor, that it runs all the way from the extracellular space to the canonical binding site with two energy minima that have to be overcome, the selectivity filter, and the same minimum was found at this point just before the canonical binding site. We ran an additional simulation in which we removed the first two ions, and this is what you can see here, and we can see that immediately the first ion really jumps into this site just before the canonical binding site, mostly pulled by this aspartate residue here, while the second one stops just behind the phenylalanine residue I've mentioned previously. So the simulations led to the identification of two additional sites, which we call the proximal binding site, close to the canonical bind, and the distal binding site just behind the phenylalanine residue. And of course, we wanted to identify more on this. So we now have identified three residues that seem to be important. There's the phenylalanine residue, which forms this constriction. There's the aspartate residue that has this energy minimum here and is important for pulling the ion all the way towards the canonical binding site. And there's the lysine residue I've mentioned previously that is important for getting the ion out of, or seems to be important for getting the ion out of the canonical binding site. But for addressing this, we did a couple of functional assays combined with MD simulations. What we always do is we measure HPase activity, we measure the transport of potassium, and we look at the distribution of the ions in a simulation. What we first did then was we exchanged the phenylalanine for an isoleucine. And the idea behind was that we would avoid the formation of pi-cation stacking, because that is what you would expect when you see a phenylalanine in a tunnel where, potassium ions propagate through. You can expect or hypothesize that there would be cation pi stackings. And in fact, what we see is that upon this mutation, we see the same stimulated HPase activity, so the same dependency on potassium, while the transport of potassium ions is massively decreased. And what we can see in the simulation is that the density that is now missing, or that is less occupied, is this distal binding site. And this is likely because the pi-cation stacking between the distal binding site and the phenylalanine is avoided because it's no longer present. Well, next we introduced a mutation in this proximal binding site, because the question was how is ADPase activity coupled to the potassium ion? We had another residue that showed uncoupling, where the I could show you already, where the aspartate residue was mutated by an alanine, which was completely uncoupled from HPase activity. So there was the idea that this proximal binding site is important for stimulating HPase activity. And in fact, when we added this positive side chain into the proximal binding site, we again saw uncoupled HPase hydrolysis in favor of our mechanism. And we saw, of course, no transport, because the potassium ion can no longer be forwarded towards the proximal binding site or the canonical binding site. And the same holds true for the simulation for the aspartate mutation. Last but not least, we also mutated the phenylalanine residue for an alanine, such that we not only removed the chance for pi-cation stacking, but also the sterical hindrance this residue would cause. And what we see now is also an uncoupled transport activity, or HPase activity, while the transport activity is again drastically reduced. And our idea of this is that in this case, in fact, we can make use of the tunnel, and we can have protonation of the proximal binding site just from the water molecules that are around, such that we see now HPase activity that is uncoupled from the presence of potassium ions. So all this finally leads us to the suggestion of the following molecular mechanism for the ion propagation. So we think that if there's potassium present, the first ion will be immediately forwarded to the aspartate, which really moves it forward due to the energy well that I've shown to you. But then also the cation pi stacking from the phenylalanine would help the coordination of this residue here. If then a second ion approaches the phenylalanine, the pi cloud is disturbed, such that this interaction is slightly weakened. And then we have a proton swap from the lysine to the aspartate residue, such that the potassium ion can be forwarded into the canonical binding site, and this is what you can see here. At this point, these two residues are not charged, which also frees the ion from behind the phenylalanine, and this exactly reflects all the structures that we have. So we see a density here, but we don't see the binding of potassium ion here or at the distal and the proximal site. And then lastly, we have the ATP hydrolysis, and the proton is switched back such that the lysine pushes the ion out of the canonical binding site. Okay, I hope with this I could show you that this is that there are in total 3 quite fascinating different families of potassium uptake systems, which only together can act to grant potassium uptake at any condition a bacterial cell could face and allow the survival of the cell. And with that, I would like to acknowledge all the people that were involved. So from all the people here are involved in this work, but the data I've showed you were from Marina Schrecker on the KTR system, Igor, Yvonne, and Michael on the KIM-A Cook family, and Charlotte and Jacob on the KDPF ABC system. I mentioned our really fantastic collaboration with Janet and Werner, and the work of Joanna, Jan Philipp, and the great help from Derek, Sonja, and Simone. We have a fantastic collaboration on the KDPF ABC system with Christina, with Lisa doing the Cryo-EM, and Gerd, who was very helpful with running the instrument. Also here, a big thanks to the MPI of Biophysics, who now allowed us to freeze our grids at the MPI such that we can continue our work. And also, I would like to mention particularly here our collaboration on KIM-A and KDPF ABC with Robin and Phil, who did simulations on both systems. And with that, I thank you for your attention.
Inga, thank you very much. So we maybe can give you a round of applause. Do we have questions from the audience or from the chat? If no, I have one. So when you looked at your KIM-A, which was inhibited and non-inhibited, maybe you. So I've seen that it seems like the transmembrane domains were differently positioned, and without the inhibition, they were bending the membrane much stronger. Is it functional, or is the effect of?
I didn't elaborate on that because it would take even more time. So the difference is how the sample was prepared. So the first structure we solved in SMALPs, and meaning we solubilized directly in SMALPs, and then we purified our protein, which probably washed out the lipid at the interface between the dimer. So in this case, the dimer toppled over. And now we purify our membranes just normally in detergent, and then we reconstitute in amphipoles upon the addition of lipids. And that apparently helps to fill up the gap such that we now get the upright dimer. And this was something we have simulated with Phil. In principle, both conformations are possible. So also in the simulations, he sees both conformations, but we believe that the more relevant one is the upright dimer. And this has nothing to do with the inhibition. So the reason you say.
Thank you, Inga. In the interest of time, we'll have to thank you once again, and proceed to the next speaker after a very, very short break. Great. We're back, and it's a pleasure to have Albert Guskov, who is the co-organizer of the conference, and so he works in the University of Groningen. He'll be talking about conformational ensemble of a glutamate transporter homologue. The stage is yours.
Thank you, Misha, for the introduction. And I must tell, I feel a real thrill actually to give finally a live talk after 1 year of those numerous talks when you just stare at your computer with some hope that someone else is on the other side listening to you. So at least now I see a few faces. You're listening to me for sure. So to save some time, let's just jump to the story. And I will tell you about the conformational ensemble of glutamate transporter homologue, which belongs to solute carrier family 1 of transporters. And why actually do we need to study those transporters? As you might know, most of the people, when they talk about the drugs for membrane proteins, they immediately think about GPCRs. And of course, there's something else beyond GPCRs. Or other people think about ribosomes because most of antibiotics work against, and they just bind to ribosome and kill the organisms. But actually, male functions of about 20% of genes coding for solute carrier transporters, they lead to Mendelian diseases. And in this slide, you see actually an overview of different transporters linked to the transport of certain compounds or nutrients. So for neurotransmitters, there are 2 certain families, SLC6A and SLC1A. And the first family, it's the family which is responsible for the transport of all those fancy things like dopamine or GABA and noradrenaline and so on. However, SLC1 family is also quite important, and it transports not such a fancy molecule, it's just a glutamate, but nevertheless it's one of the most important neurotransmitters available in our bodies. So SLC1A family of transporters, it's a relatively small family, so it has only 7 members, and 5 of those members are excitatory amino acid transporters, and 2 extra members, they are involved in that exchange of neutral amino acids. And most of the studies actually, so you can see on the slide also the structures popped up, the first structures pop up. So up till 2017, all structural work has been done on the archaeal homologues. And you may argue actually whether it's a good study organism to study these transporters, because if you pay attention, they share only about 25% of sequence identity, which is not considered very high, as you might know. So the lucky thing is that the conservation at the subterminal domain is much higher, and that's the domain where actually all the residues necessary for the transport of substrates are located. And it actually turned out not to be a very bad and perhaps even excellent model to study many things related to this family of transporters. You don't really have to go through all this slide. It just all the diseases known so far which are linked to malfunctions of these transporters. I will just mention a few. So, for example, schizophrenia or impulsive obsessive disorders. So you can already feel that, well, these transporters are quite important. So what actually they do is that they pump back the glutamate, which is released into the synaptic cleft. So whenever you have the transport of the signal needs to be transported, so glutamate is stored in vesicles and then it's released, and actually then it ends up on the glutamate receptors. So then the job is done, but it's better to remove the glutamate as soon as possible. Otherwise, you will have an overstimulation, which is not a good thing. There are also certain conditions, like ischemia, for example, then gradients of ions are broken, and these transporters start working in reverse, and that actually leads to neurotoxicity, and some neurons may even die. So not a very good thing either. And also glutamate was actually shown quite recently as a strong indicator of brain cancers, of gliomas. Because what happens, so you see it on the right, that, yeah, you should see it now, that in gliomas, they require a lot of cysteine to synthesize glutathione to withstand the redox stress. So they actually kick out glutamate as much as possible, and they do not express the AT transporters. So you actually get, well, a lot of intoxication due to glutamate. So if you compare human transporters and these archaeal homologs, they are pretty much alike. There are few certain differences, like, well, the most important is that in human transporters you have a co-transport of proton and counter-transport of potassium, and these transporters are much faster, and they really do transport glutamate, whereas archaeal homologs, they rather prefer aspartate, and they really use it as a nutrient. So we started studying this family extensively almost like 10 years ago now. And well, back in 2013, we actually characterized one of these transporters from a certain archaea, and we showed that this is indeed aspartate transporter, which is sodium dependent, so you don't have any substrate binding in the absence of sodium if you, well, look at the ITC profile on the right. So when you have zero sodium, you have, well, zero binding of aspartate as well. So there were numerous crystal structures from different groups. From Olga Budker, she made an amazing work actually to decipher first structures of these transporters. And people realized immediately that they are trimers, and they actually consist of two domains, so a scaffold domain and transport domains, and those domains, transport domains, are located at the periphery of a transporter. And in 2013 and 2016, we solved structures of this transporter in two different states. In the upper state, which is completely empty, so void of anything, no sodium ions, no substrate, and fully substrate loaded. And, well, do pay attention to the position of at least one amino acid here, which is methionine. And we use this position as a very strong indicator whether the transporter is in apostate or in substrate bound state, because it really swings like about 9 angstrom or so. So just remember for now, because it will be important later, that in the apostate and in the substrate bound state, there is a huge difference in the positions of amino acid side chains, which do actually interact with sodium ions with a substrate. So we also showed a few years later that these transporters can transport and bind both stereoisomers L and D, but I will not well, tell you any more details about that because it's rather irrelevant right now. And there's been really tons of studies on this transporter, not only from the structural biology, but also from single molecule thread, from EPR, from AFM, biochemistry, mutagenesis, and so on and so forth. And what we noticed, actually, all the data, they point out that this transporter operates in this elevator-like mode, and these elevators, transport domains, they operate independently. However, all structural studies actually almost always showed that these transporters are parked at one position. So either outwards, then all of them are above the membrane, or inward, then they below the membrane, or somewhere in between, intermediate. But they always sort of in the very symmetrical arrangement. One of the problems could be that all these studies were performed in detergent, and well, we already know from several talks during this conference that detergent is not the best way to isolate and study your protein, especially if it's a membrane-embedded protein. So we moved to nanodiscs, and we were actually bought by this MSP2N2 by its size, so it fits well, very nicely actually the size of target protein. So we thought, well, that would be cool. So we just reconstituted, and we have lipids around, so protein should feel happy. Well, it actually felt happy, so I will not go into details of all the preparation, but we just reconstituted it with lipids and with nanodisc with belt proteins. And then using actually our in-house Arctica microscope equipped with K2 camera, we collected quite a lot of data at different conditions, so I will not go into details of this EM pipeline right now. I will just tell you that all processing was done using CryoSpark. And what we got at the end. So here you see kind of titration of the substrate to the protein. So we start with, so first of all, we used the same amount of sodium in all conditions. And when we started to titrate the L-aspartate, so we really started with 0 aspartate. When we add some, just to have it in unsaturated state, and then we saturate it fully. And you can actually see that at fully saturated state, which is on your right, the positions of transport domains, they are in symmetrical state. But those which are squared, I believe, yeah, you should see it in rectangle. So those are very interesting because they're in unsaturated conditions, and you have actually 2 populations. So you have a population of a protein where you have 1 transport domain in the inward state and 2 outward state, and the other way around, so 2 inward and 1 outward. And actually also the first sodium-only state is also very interesting, and I'll show you why. So you may also wonder, why do I have an extra one just below with TBOA? And TBOA is a known inhibitor of these transporters and actually used to lock the transporter. So there's a certain gate of the elevator. I'll tell you a bit more about it in detail later. And actually, if you put this inhibitor in, so you, well, you block the transport. So the transport domain cannot move, well, down or up. So important thing to mention that this nanodisc, the belt protein, is not very well resolved. So it's sort of a bit heterogeneous. And you can see that the resolution, well, of a protein is pretty good. So it's around 3 angstrom in the core. But at the periphery, it's getting, well, really poor in some cases. So now we have a very complex table in a way, but actually, if we look at those transport domains, we can classify them according to different things. So first of all, we can classify them according to their position. So as I mentioned already, if they above the membrane, so it's called outward state. If they're below, it's inward state. They can be also an intermediate state, of course. But also we can say whether the transport domain is loaded with substrate or not. So it can be hollow state, and then it's aspartate or an inhibitor. It can be sodium-only state. Then it's only sodium, but no substrate present. And also, since I mentioned there is this certain gate leading to the binding site, so that gate can be open. And when we talk about open state of that transport domain, or occluded. So when we have a bound substrate and the gate is closed, and actually it's the only situation when domain can go down or up. So if this gate is open, it will not be possible for the transport domain to move. So how did we assign those states? And actually, this is where our previous structures we got came very handy. So here you have superpositions. Well, between different states, we resolved using Cryo-EM, and in gold, we have a gold standard crystallography models, which we obtained earlier with relatively high resolution. So you can actually see in case of sodium-only states, we have a very good correlation with positions of sodium we determined earlier. And in cases which are apo, so we don't have the sodiums present, we actually don't see any density for those. So that makes us, well, very confident, and especially, as I also mentioned, the positions of side chains matter a lot. Actually, we can quite easily deduct whether we have apo state or substrate-bound state or even sodium-only state. Because basically the easiest way to say whether we have substrate bound or not is to look at the density, at the corresponding density, and in case we don't have substrate present, we don't have corresponding density, of course. But the important thing that the transporter is primed for the binding of a substrate. So all the positions of amino acids which are needed to bind substrate are already there. So it's just ready, it's just waiting for the substrate to bind. So I've mentioned already about that gate which shields off the binding site, and we could actually resolve, although we already seen that with a different member of this family, namely ACT2, that HP2 is the sole gate necessary for the transporter to operate. And the conundrum was that next to HP2 there is also HP1, and they are pseudosymmetrical. And the initial idea, which is actually very obvious, was that, okay, HP2 operates in the outward facing state and HP1 would operate in the inward open state. But, well, surprisingly, it's not the case. So only with HP2 you can efficiently, well, both load and unload the substrate. So coming back to TBOA structure. So the interesting fact, we observed that in our EM structures without TBOA, all the positions were not fully outward. But to be able to bind the substrate, actually it should go all the way up. And using this inhibitor, we actually showed that indeed in our conditions, in our setup, transporter can go to that position to pick up the substrate. And actually, if you look at this slice through now, so the transporter domain, which is in outward position, which is up, and it has a bound inhibitor, and you look at the position of inward transporter, which is with open HP2, with sodium-only condition. So you can see that this is how transporter prevents that the transport domains move without substrate. So as soon as the HP2 gate is open, the transporter cannot operate anymore. So you really need either to bind the substrate to operate, or you need to kick out all the substrates, including sodium ions, for HP2 to close, and then transporter can reorient. And actually, the degree of this opening of HP2 is also quite easy to monitor. So, well, there are different shades of blue, which is probably a bit hard to see now, but the one which is fully open, so this is in case of TBOA, and the one which is fully closed is with substrate bound in the occluded state, and one in between is an inward open state. The interesting observation we made, which is actually relevant to many of you who is going to study membrane proteins reconstituted into nanodiscs, that the size actually differs quite a lot. So you remember I mentioned that we actually decided to use this MSP2N2 protein because of the size, because, well, it's on average between 15 to 17 nanometers, but judging from the structures we got, it is actually around 10 to 11 nanometers only. So keep that in mind. So most possible scenario is that upon your read preparation, this nanodisc actually shrinks quite significantly, and it may even squeeze out the lipids, which you actually want to keep. We also estimated the bilayer deformation. And while we noticed that in the areas where transporter goes down in the inward facing state, we have most severe membrane deformation. But can we improve the resolution any further? Because, well, of course, you've seen those beautiful resolutions from previous talks where people have 2 angstrom and so, and we are relatively stuck with 3.4 because we have only 200 kilovolt microscope without any fancy cameras of new generations. And we actually can do quite some improvement. And what we did, we actually used this not so new program, but well, relatively new, TOPACE. So I will not go into details, but there are publications mentioned in the corner which I really recommend you to read. So it's actually a neural network-based program which you can train, and then it can do a better picking for you. So we did it with a few iterations, and actually at the end we managed to improve the resolution going from 3.4 almost to 3 angstrom, and it really matters. Because of course we want to see also the lipid positions. And well, I already mentioned that those nanodiscs are not so big, and we were really afraid that none of the lipids are left. And fortunately, there are a few of them, and those are located sort of in the crevices between the transport domain and the scaffold domain. And actually, if you look into detail, they are not so far from that gate which opens and closes to load and to unload the substrate. So perhaps those lipids are important for the transport function of this transporter, but well, this still needs to be investigated. So with that, I would like to conclude, and hopefully I saved some time, I don't know. So we actually showed, so maybe the most important take-home message for you, so if you're gonna work with nanodiscs, so keep in mind that they probably shrink when you freeze your grids. So I showed you today that this transporter actually indeed works in the asymmetric mode. Those transport domains, they move independently. And well, you can use only one gate for this transporter. Why it's like that, nobody knows. So lipids probably important, but well, we still need to investigate their role, and we still need to find a way how to keep more lipids around it. So there are plenty of people involved who were involved at different stages during those past 10 years or so. But I would especially like to mention those people on the right. So there is Valentina, who was a postdoc with us for many, many years, and now she's a research scientist at Zobio company. So Valentina represents here an inward open state. So Professor Slotbaum, who is my friend, mentor, and colleague, well, he's, well, if you pay attention, he's holding a bottle of champagne in his hand. So he represents a TBOA-bound conformation, also with an open HP2 gate, and well, I'm actually showing the intermediate occluded state if you look at my face. So there are also a few people who are working hard now, continuing this project, and those are my PhD students, Emanuela Colucci, Maria Nemchinova, and Igor Marin. Well, I thank all of you for your attention. I'll be happy to answer your questions if you have any. Thanks.
Thank you, Albert. We have a vivid discussion in the chat here. So Leonid Sazonov says that he found that nanodiscs can be not only smaller, but also larger than predicted, depending on the protein size.
I fully agree, because we managed once to put photosystem on those nanodiscs.
Okay, excellent. So there is also a question of what is the diameter of the reconstituted complex, if you measure the actual number.
So I believe the size of a protein, right? Yeah, so it depends how you measure. It's around 10 to 12 nanometers.
And what's the size of the nanodisk, like the textbook size that was supposed to be?
So it's between 15 to 17, depending on the technique you use, so size exclusion of SAGs.
Okay, thank you. More questions? I also have one. So obviously you have different state of your protein depending on the strength of your ligand, right? On the concentration.
Yes, exactly. Yes.
So, but the way you picture it is that at a certain concentration there is only one preferred conformation. I would assume that there would be, for every condition, you would have a conformational spectrum where one state maybe would be more preferential, but the other states would also be present.
Yeah, so they're probably present, but let's say at very low population, because apparently there are certain energetically favored states, and let's say with the bound substrate, it tends to stay symmetrical for whatever reason, just because probably it's more stable.
Mm-hmm. Yeah, very interesting. Okay, one more question. Do you recommend that MSP2N2 shrinks to accommodate the complex?
Well, I cannot recommend it. I just observe it. So
okay, good.
I don't get the question though. I just be careful with that. I mean, perhaps it indeed really depends on your protein, but don't be surprised, because idea we had is to have many lipids around, but apparently all of them just got squeezed out.
Okay, thank you. We'll thank then the speaker and all the speakers from this session. So let's put a round of applause. We will have a 10-minute coffee break now, or a tea break, and we'll come back at 10 minutes past a full hour. Okay, we're back online. We're very excited. So it's a pleasure to have our next speaker. That's Anna Protopopova. And so she is from University of Pennsylvania, from the group of Vera Moisenkova-Bell. Anna, do you hear me? Okay, we cannot hear you so far. I think you're muted.
Can you hear me now?
Yeah, excellent. So Anna will be talking about modulation of TRPV2 channel by small molecules. The stage is yours.
Thank you. First of all, good afternoon, everyone. It's a great pleasure to talk at Moscow State University today. I spent about 10 years of my life being first a master's student and then a PhD student there. So it's really exciting to share my current work, even if only remotely. So my talk is going to be about TRPV2, a transient receptor potential vanilloid 2, a channel belonging to the vanilloid subfamily of the transient potential receptor channels. So TRPV2 is related to TRPV1, TRPV3, and TRPV4 channels. Together, those 4 channels are sometimes called thermo-TRPVs because they all have some level of temperature sensitivity. And on the first day of the conference, you have heard a talk by Alexander Sobolevsky about temperature activation of TRPV3. And today I'm going to continue on a closely related topic. So TRPV2 is a ligand-gated non-specific calcium channel. It is expressed in most tissues and organs of the human body, including the brain, the heart, the peripheral neurons, the white blood cells, the pancreas, the bladder, and the prostate. And multiple studies have shown TRPV2's role in both healthy and malignant tissues and its effects on progression, drug resistance, and metastasis of different forms of cancer, making it a promising target for drug discovery and also just an interesting object to study. Structurally, TRPV2 is a tetramer. Each monomer consists of, each monomer has an extensive cytoplasmic part, mostly consisting of 6 ankyrin repeat domains, ARDs. Also, the N-terminus and C-terminus of each monomer is in the cytoplasm, where they have been proposed to have an important regulatory role, and I'm going to talk about it later. Extracellular part is small and mostly unstructured, and then finally, each monomer has 6 transmembrane helices. Helices S1, S4 form a helical bundle. A short helical linker, S4, S5 linker, connects the S1, S4 bundle to S5. S5 has 6 and short pore helix between them, together called a pore domain, reach out to the neighboring subunit, creating the central pore of the channel right in the middle of this tetrameric assembly. Overall, thermo-TRPV channels, TRPV1, TRPV2, TRPV3, and TRPV4, all have very similar architecture. The gating cycle of TRPV2 resembles the gating cycle of many other ion channels. Its resting state is closed and does not let ions to go through the membrane. When we purify channels to freeze and solve their structure without any added ligands, we are getting so-called apo state, which we believe in case of TRPV2 is a resting state. Then upon application of a certain stimuli, the channel is getting activated and opens, but it does not stay open for the whole duration of stimuli application. Instead, it transitions to the closed state, which is different from apo state and called inactivated. Again, all thermo-TRPVs seem to have similar gating cycles and perhaps even driven by similar structural mechanisms. V1, V2, and V3 have been studied extensively by multiple groups, and a lot of good ideas were proposed on how those channels can open and inactivate. But until now, the wild-type full-length channels never gave an open state. And it always required mutations or truncations or a certain combination of both to open those channels. And the goal and the strategy of our studies in Vera's lab is to try and use native system to see all the stages of the gating cycle. So on the biochemical level, we always try to use wild-type channels. Luckily, the strategy works for TRPV2. Today, I'm going to be showing all the results from wild-type, full-length, no mutations, rat TRPV2. Then we reconstitute our proteins in nanodiscs to try and mimic the cellular membrane environment as much as possible. And then we also using a known activation stimuli to open the channels. And the stimuli could be heat, temperature, as you have seen in Alexander Sobolevsky talk on the B3. Heat is probably a challenging one, especially for TRPV2 because it has a very high temperature sensitivity threshold. We mostly use small molecule ligands that can open the channel. On the level of sample preparation and data collection, we do our data collection in a classic way. We freeze on holy carbon grids, and TRPV2 does have a strong preferred orientation, so we have to add the detergent for better ice thickness and better angle distribution in our particles. And therefore, after the addition of detergent, we are left with not that many particles inside the holes, though still plenty, but we have to collect large data sets to get to high resolution. And last but not the least, we have seen in many data sets, and not with TRPV2 only, that data processing has to be taken very thoughtfully and carefully, because almost every data set that we got had some sort of heterogeneity of different nature. So it has to be very, very careful process. And we're always trying to get multiple states from the data where possible to get the whole picture of what's going on in the sample. So coming back to biochemistry, for today, there are several known exogenous modulators of TRPV2, activators, inhibitors, and pore blockers. We are trying to understand how all of those compounds modulate TRPV2's activity. But today I'm going to focus on only two of them, an activator 2-APB and the pore blocker ruthenium red. The apostructure of red TRPV2, so a resting state, has been solved previously in nanodiscs in our lab using Cryo-EM. Turned out that TRPV2 has two apostates, with the only principal difference between them at the pore. So if we look at the transmembrane part of two opposing monomers, we will see a pore profile. The dots here in the middle of the pore are showing the solvent accessible area inside the pore. And as you can see in the pink APO1 state, there are two closed gates. The lower gate at the bottom is tightly sealed with histidine 651 and methionine 645. The upper gate, called selectivity filter, is closed with this whole loop, part of the pore loop. In the purple APO2 state, the lower gate remains closed, but the selectivity filter is open. The loop completely went out of the way, away from the pore. So the selectivity filter of TRPV2 naturally has some conformational flexibility. It does not function as an actual gate and can open spontaneously in its resting state. These results, published from Vieira's lab in 2019, are in a very good agreement with the electrophysiology paper from Kenton Schwartz group, published, I think, on the same year, where they shown a little more generally that in V1, V2, and V3 channels, selectivity filter doesn't seem to be a real gate. So with this introduction, I can now move on to our recent and still unpublished data. We wanted to solve a structure of TRPV2 in the presence of an activator 2-APB. And here you can see a single channel electrophysiology data illustrating the gating principle that I mentioned earlier. So in this experiment, a single channel in a small patch of cellular membrane is getting activated by 100 micromolar 2-APB, but it does not stay open all the time. Instead, it goes back and forth between closed non-conductive state and open conductive state. And the opening probability is actually not very high, so we expected to get definitely multiple states from the 2-APB dataset. When we began processing the data, we realized that we are not able to get discrete open-closed states, but instead we see a lot of heterogeneity in the data. And so we applied a new, recently became available, this tool only became available recently, the 3D variability tool from CryoSpark. This tool is designed for identification of continuous movements in the data, and in C4 symmetrical form of movement, we actually saw a nice channel opening. This movie is still at low resolution because this analysis is performed early in the processing, but every frame of the movie is made of actual particles. Some particles contribute to the more open states, some particles contribute to the more closed states. So we isolated the most widely open particles and the most tightly closed ones and refined them separately to obtain two states. An open state shown in teal color. It didn't have many particles, so the final resolution on this state was about 4 angstrom. And the most closed state with a resolution of 3.5 angstrom. It had many more particles. I'm going to talk about the difference between those two states in a few minutes, but I want to start by showing the 2APB binding site. So in both structures, open and closed, we were able to identify a characteristic shifts in the position of phosphorus 5 linker, which were triggered by the drug, and a new non-protein density, which is shown here in orange and corresponds to 2APB. So 2APB binds at the interface between two monomers, between the S4 S5 linker of one monomer shown in gray and the bottom of S5 helix shown in cyan. At this position, the drug is coordinated with 4 side chains. In APO structure, the same side chains form a network of connections stabilizing the interface between two monomers. However, 2APB seems to disrupt this network, wedge two monomers apart. And to validate this binding site, we first used computational approach, Rosetta docking simulations, which confirmed the overall binding site and allowed us to position the drug a little more carefully and confidently in this binding site. And then we turned back to experiments and used electrophysiology on HEK293 T cells overexpressing wild type of mutant RET2V2. In those experiments, an electrode is connected to the membrane of the whole cell, and increasing concentrations of 2-APB are applied to the buff solution surrounding the cell for 20 seconds each, with buffer washes in between. And we mutated all 4 residues of the binding site, but the most interesting results were obtained with this histidine 521. Histidine 521 mutation completely abolished channel activation with 300 micromolar 2-APB and significantly reduced the response to a high dose 1 millimolar 2-APB concentration. So therefore, we concluded that histidine 521 is the key residue determining activation of 3V2 with 2-APB. It's very interesting because in 3V3, which is also activated by 2-APB, there's a different binding site, but also the key residue is histidine there. Now, I want to show you the difference between the two 2-APB bound states. So first, the pores were quite different. The graph on the right shows similar thing as the dots here, but sort of more numerically. It shows the radius of the pore, and the dotted line marks the radius of dehydrated calcium ion. So as you can see, both APO and inactivated structures, pink and cyan, are closed at both lower gate and selectivity filter, comparing to the dehydrated calcium ion. But the teal green open structure will allow a partially dehydrated calcium ion go through the pore. The opening of the selectivity filter here is similar to the opening in the APO2 state. The loop goes away from the pore. At the lower gate, very interestingly, the alpha helical S6 helices moved apart a little bit, making space here, but also histidine 651 side chain rotated out of the pore by almost 180 degrees. And at this new position, it was stabilized by the interactions with lysine 531 at S4S5 linker, and it remained stabilized and remained in an outward position even in the inactivated state. So it's a very interesting mechanism of lower gate opening. But what really allowed us to identify the physiological meaning of our states is the cytoplasmic part of the channel and the C-terminal regulatory switch, originally identified in 3v3 by the Lee group. And it has been proposed that the open state should have a C-terminal helix near the beta sheets region here, while inactivated state should have a unwrapped C-terminals and a loop wrapping around the beta sheets. This is exactly what we see here. And very interestingly, our apo state also has a C-terminal helix. So it's sort of pre-primed, ready to open state. And this is true for both APO1 and APO2. So both resting state of TRPV2 are pre-primed and ready to be opened with an activation stimuli. Now, upon binding of 2-APB, the actual opening of this pre-primed state is achieved through rotation of the entire protein, except the pore domain, which is S5, S6, and pore helix, around the central pore axis by 7 degrees. This large movement of the entire channel originates from the drug binding site and from the drug shifting the S4, S5 linker outwards. On average, S4, S5 linker shifts away from the central pore axis by 0.8 angstrom, making space for the bottom of S6 to open. And also pushing the S1, S4 bundle to outwards and to rotate the whole assembly of cytoplasmic part. On the pore domain, again, the drug pushed S5 a little bit up, and this translated to the upward movement of pore helix by 1.4 angstrom, which allowed the selectivity filter to open in a manner similar to APO2 state. Inactivation involves backwards rotation of ankyrin repeats and the whole protein assembly by 7 degrees, downward movement of the pore helix and the whole channel. And additionally, there is a significant downward pivoting of ankyrin repeat domain. So the whole ankyrin repeat skirt sort of goes down very significantly, dragging the channel, and it looks like it's this pulling from the cytoplasmic part, which helps narrow the transmembrane region and close both gates. Finally, to get back to the resting apo state, the channel has to expel the drug and relax the remaining shifts, which is mostly the RD pivoting, which has to come back and release the transmembrane part to get a little wider, come back to its normal resting position. So this video is just a short summary of the shifts that we see, transitioning to teal open structure, and then the C-terminus unwraps, wraps around the beta sheets, and we get inactivated structure, again, closed. On the last 2 slides, I will be talking about a different way to inactivate the channel through the pore block. Ruthenium red is a component of histochemistry dye, and it's a very effective non-specific pore blocker. On this whole cell electrophysiology trace, you can see how the current evoked by 200 micromolar 2-APB rapidly stops after addition of ruthenium red. And in this case, the Cryo-EM data set was analyzed in Reliant and yielded 2 already known states, APO1 and APO2. The states were exactly the same. And APO1 had no additional non-protein densities, but APO2 was much more interesting because it had an additional density in the pore, shown in red here, just below the selectivity filter, which we identified as ruthenium red. The ruthenium red binding site is located between the pore loop and the S6 helix, where it seems to be coordinated by several carbonyls on the pore loop, tyrosine 634 inside the pore, and glutamic acid 609 on the pore loop. If we look at the pore profile, it's clear how ruthenium red can work as an effective pore blocker. Molecules of ruthenium red sitting here below the selectivity filter close even the open selectivity filter, and ruthenium red is also positively charged, so probably the upper gate becomes absolutely closed for ions, no matter what's happening at the lower gate. To validate this binding site, we again used electrophysiology experiments on HEK293 T cells overexpressing wild type and mutant RAD3V2. We tested glutamic acid 609, which is a negatively charged residue, and we mutated it to glutamine, a residue of a similar size but uncharged. As a control, we used a nearby glutamic acid 599, which is located on the pore helix. And as you can see, glutamic acid 609 mutation significantly affected the channel conductivity in response to 2-APB, but it also almost entirely prevented current inhibition with ruthenium red. And both responses remain normal in the control mutant. Therefore, we concluded that glutamic acid 609 is the key residue determining ruthenium red block of TRPV2. And this residue is also conserved in V1, V2, V3, and V4 channels. And this mechanism of the pore block seems to be conserved for all terminal TRPVs. Together, our results allow us to propose the model for TRPV2 gating, in which the TRPV2 selectivity filter is not a gate, but instead it has a conformational flexibility to allow for the entry of large positively charged pore blockers, such as ruthenium red. Upon binding, ruthenium red blocks the TRPV2 pore at the selectivity filter through the interactions with the carbonyls of the pore loop and the side chain of glutamic acid 609. The binding of 2-APB at the interface between S4, S5 helix of one monomer and S5 of an adjacent monomer leads to channel opening and subsequent inactivation with C-terminal regulatory switch. And histidine 521 is the key residue of 2-APB binding site. With that, I want to finish my talk and say thank you to everyone who was involved in this work. First of all, Vera and Ruth. All the work that I presented is done together by me and Ruth. We worked together on this project, and I'm relatively new to the field, so actually I learned everything I know about cryo-EM and structural biology from Ruth. Then I want to thank people from Andreas Leffler group at Hanover Medical School. They performed electrophysiology experiments for us, and a group of Vladimir Yarov-Yarovoy who performed Rosetta Dawkins simulation for us at UC Davis. All the members of Vera's lab and Cryo-EM and electron microscopy course at Penn. Thank you for your attention, and I'm happy to take questions.
Anna, thank you very much. Maybe we can give Anna a round of applause again. Yeah, thank you. We have a question in the chat. So one is about if you use 3D variation analysis in CryoSpark in a cluster mode in order to get separate particle groups for different states.
So the separation of particles based on this 3D variability, I think it was not a very straightforward process, but the particles do get some attributes that you can use later on to basically manually, not one by one, but sort them out manually.
Mm-hmm. Okay. Yeah, interesting. So I have a question myself as well. So you don't see ruthenium red, the density for it in your pore, is that correct?
We do see a density in the pore.
Oh, okay.
I think I don't have a good image here. But it's right between the monomers. It's a pretty big density, significantly bigger than what is present in the APO2 structure, which is
okay. So there is one molecule that's enough to block the pore.
We see 4 molecules, and even when we refine without symmetry, we do see 4 molecules. We are not entirely sure that the channel is actually blocked by 4 molecules, so it's just sort of misalignment. We don't know. We don't know the stoichiometry. We do see 4 molecules.
Mm-hmm. Okay, and then I also had a little question: if the activation process has any asymmetry component to it, because everything I've seen looked like it had C4 symmetry.
Yes. So we were specifically interested in C4 movements because the channel is C4, and what was previously seen for V1 and V3, the open structures were all C4. There is a lot of asymmetric movements, and there is all sorts, basically. There's C2, there are asymmetric movements. We were not able to pull any asymmetric structures in good enough quality to get the structure. And we honestly don't think that they are physiologically relevant for activation. Maybe they are steps in inactivation, if that makes sense.
Thank
So
you.
the channel first rotates to open, and then one by one, the subunits may be transitioning
Interesting.
into this state.
Interesting.
But we were not able to get good structures out of any of the asymmetric. No.
Thank you. More questions? We don't have any in the hall and none in the chat. Thank you very much, Anna. So thank you for your talk, and let's thank Anna once again. We will come to the last talk of our symposium in a second. Stay with us. Yeah, so unfortunately we had technical difficulties with connecting to the last speaker, and we will now proceed to the concluding remarks for our conference. We really enjoyed it, and so to close the conference we have Olga Sokolova. Please go ahead.
Okay, thank you, Misha, for introduction. And dear friends, unfortunately, this is end of our conference, so we finally came to the end. And as you see here on my presentation, this year is the year of the science and technologies in Russia. So we were just in time with the science and technologies. So our conference was from May 30th to June 2nd for today. And let me just go a little bit back to in the time to see first the conference was held in 2017. We only have 120 attendees, 27 speakers from 8 countries. Next conference was in 2019, and we already made 200 attendees, 40 speakers from 13 countries. And this year, due to pandemic and due to a lot of people who were online, we have actually 600 attendees. 500 from them were online, 44 speakers, and 35 were online from more than 25 countries and more than 150 cities. I was trying to calculate the cities and I actually get lost. So we have attendees from all over the globe, from USA, from Japan, Russia, Israel, France, and a lot of different other countries. Also yesterday there was an online poster session with I think was also a great success. We have 30 posters and very successful online discussion. So actually the idea of making the hybrid formation of our conference, I think it was a very good idea, because people who could not attend, they can come and see what's going on and discuss. And our people who were our young scientists, they can have some advices from much more mature scientists. So now to the main scope of the conference, and I think that I would not be mistaken if I will say that its main scope is a dramatic increase in the resolution. So, and then you can, if somewhere like 50 years ago, all proteins were drawn by means of rectangular and circles, like Yuri Pelikhanov today mentioned. Today, we just seen a beautiful image of ATP synthase, and we all like this red and yellow model of ribosome that was presented in the 70s of last century. And then here, now we can see beautiful reconstruction from several labs. And also, so all these great resolution, now it's possible to visualize different conformational changes. It's possible to visualize water molecules and also hydrogen molecules. So now Cryo-EM is on absolutely new level. So what we need to tell also is about like current situation with cryo microscope, that as Carolyn Luger told us that there's a great lack of electron microscope in the center of United States. So, but if you can see European part of center of Europe, we actually can see very similar lack of the microscopes. And actually this diameter of this circle is very similar. I try to estimate. You can see here we have a scale bars. So it's just estimation, but the Here we have only one microscope in Moscow and then a lot of microscopes in Europe. But actually, if you can see, if you imagine the whole Russia, then the circle would be much, much larger. So, and we only have one microscope in Moscow now. So that's why we're going again to current situation with cryo microscopes. And these figure I showed 2 years ago. What was at that time in MIPT, there was no microscope. There was microscope in place of MSU microscope. There was a microscope in St. Petersburg, but people in St. Petersburg don't really use Cryo-EM. So what now we have in 2021, we got a grant from Ministry of Science from Russia, and we upgraded our GEOL 2100 microscopes to make it cryo microscope, and also MIPT installed their Polara. So now we have several microscopes where we can check our grades, but we still have only one good Titan cryos in Kurchatov Institute, which is not it's not getting younger with the time. So it's already 6 years old. But still, despite we only have one microscope, we have some reconstructions obtained in Russia with the means of this microscope. And so you can see it's a GPS DNA complex and a ribosome and a membrane proteins, and also beautiful chaperonins that Evgeny Pichkur demonstrated yesterday. And these, I should draw your attention to the structure of base plate of the bacteriophage and the capsid structure of the same bacteriophage that were solved in my lab, and they have been submitted to as the poster session yesterday. So even we only have one microscope in a very big country, we still are working, and young people are motivated. So that's why I believe that we need to do something to change the situation. So we need to suggest the resume of the conference. So we're suggesting, we've been talking to some people in during our discussions offline here at the conference. So, and what we suggesting, we probably should provide a letter to the Russian Ministry of Science to support our initiative to form a national center for collaborative use of cryo-electron microscopes, because we really, it's very few microscopes for such a big area, and for so many people who wanted to do structural biology. So here I'm coming to acknowledgements, and I think again everyone will agree that we have a great conference. So I've been talking to different people, and everyone like our conference, and this is all due to great program committee. So this is our great program committee. It includes Yelena Orlova, Natalia Deval, Alda, Konstantin Boyko, Albert Guskov, Andrei Konivega, Evgeny Pichkur, Andrei Rogachev, and Konstantin Usachov. So all these members of program committee, they talked to the people, they invited different people, and helped us to build this great program. And also the technical support. Again, so Elizaveta Trifonova, Dmitry Pleshkov, Andrei Bershatsky, and Polina Kalinina, who wrote the letters, tons of letters to speakers, who design all our website and everything else. And without this very experienced technical support, we could not do anything. And also our students who volunteered at the conference, they were sitting at the registry, and they were talking to our speakers on Zoom. And this is Yekaterina, Daniil, Daria, Nadia, Naida, Yekaterina, another Yekaterina, Alina, Ilya, and Sophia. So very thanks to student volunteers. Also to our sponsors, which are at the bottom, and International Journal of Biomedicine provided just today they published the supplement where all accepted poster topics and texts were published. So you should also be aware that conference photographs will be published on our website, maybe a little bit later. And thank you for your attention. Now we are looking for the next Russian Cryo-EM conference, which will be in 2023. So we really looking forward to this conference, and we're really looking forward to make it fully in person. So we invite all potential speakers in 2 years to Moscow. So we hope it will be in Moscow University, and everything by that time will be good. So now with the final words to the conference will the director of Federal Center of Biotechnology, Alexey Fedorov, will talk, say some words to our conference.
So we're back online, and to close the conference with a final word, I'm happy to introduce the director of the Federal Research Center for Biotechnology, Alexey Fedorov. Alexey, please, the stage is yours.
Okay, thank you. First of all, I'd like to say that I will make just a few final remarks. I think it was truly an extraordinary conference with a lot of insights in this so fastly developing field where cryo-electron microscopy allows to solve so many problems in really many different experimental directions and settings. So I'd like to thank all the speakers for their great job for explaining for us state of the art and giving us so much food for thought. I'd like to thank, as Professor Sokolov already did, to thank organizing committee for their also, it's not very easy job organizing any conference, but under these particular circumstances, which we all understand, it's not an easy task at all. We on our side, I mean Federal Research Center of Biotechnology and Moscow State University as host organizations, were trying to do our best to make this conference really productive and comfortable. I'd like also to thank sponsors, of course, and also one also very important point is that at the very first talk, there was a talk on making these tools and devices for cryo-electron microscopy more affordable, or as the speaker put it, to make Volkswagen Beetle along with the Cadillac, very important thing. And what makes it maybe major step from just wishful thinking from our side as a research center for real consideration, maybe of buying one or another piece of such equipment for using a cryo-electron microscopy. So I'd like to thank again all the organizers, speakers, and attendees of the conference, and hope that we all can make it at the next conference in 2 years from now. Thank you all.